dito din live once.
Ay, nag-livestream sila sa YouTube.
Hello.
Hello, sir. Good morning.
Hello.
Hello, sir.
Good morning. Yes.
Good morning.
My voice. Abhishek. Hello, Abhishek.
Abhishek? Abhishek?
Is it audible?
Yes. Yes, sir. You are audible, sir.
Am I visible to you? Yes.
Good morning, sir.
Attendees are how many? 39 attendees are there. Okay.
Abhishek, am I audible?
Yes, ma'am.
ये आधा स्क्रीन देखने वाला है। थोड़ा इसको करो तो but for this is तेरे को क्या चाहिए एग्जिट फुल स्क्रीन कर क्या हो रहा है Yes, sir. Good morning, sir. I can see you now.
Yes. Thank
Welcome,
you.
sir.
Great, great. Nice to see you.
We are fortunate, sir. We are fortunate
Me too.
to have you with this e-conference.
Yeah, me too. Me too.
Exactly, sir. Today is our holiday for Guru Nanak Jayanti.
Oh, okay.
So that's why we all are from our own home,
Okay, okay. No problem.
not from institution.
Good, good. So you are continuing online.
Yes, yes. It is online and offline both. But last day only it is in blended mode. Today we are in online.
Okay.
Sorry, your blessings.
Best wishes are always there. Blessings ऊपर वाले का.
Also, you people also have, sir. So, Khushbu ma'am,
Yes, sir.
should we start? I think this is the right time to start the event, please.
Yes, sir. Yes, sir. The new day brings new hope. The lives we have led, the life we have yet to lead. A new day, new ideas, a new you. A very good morning to everyone. I, Khushbu Raj, Assistant Professor, School of Pharmacy, Arkajan University, welcome you all to the second day of International Conference on Natural and Novel Pharmaceutical Research, collaboratively organized by School of Pharmacy, Arkajan University, India, and Department of Pharmacy, San Pedro College, the Philippines. Now, we will begin our today's plenary session with our speaker, Mr. Ranjit Vaishkar. Good morning, sir.
Good morning.
He is a CEO of QBT International United Nations Advisor Geneva. Sir has also worked as quality expert with World Bank Washington, USA, and with Saudi Arabia FDA. Apart from that, he has also worked with various pharmaceutical companies and labs such as Pfizer Limited, Sun Pharma, Leica Labs, Cancer Research Institute, Mumbai, and he was retired as Global Quality Head in 2006. He has also delivered 498 presentations in various conferences and approx 60 webinars till date in last 14 months on COVID and various pharma subjects. Also, he has various awards in his credits like Quality Champion Gold Award, Pharma Ratna, etc. We welcome you, sir, in on the second day of our international e-conference. Now, may I request you to kindly enlighten the session with your knowledge. Over to you, sir.
Thank you very much, Dr. Jyotirmay Sahu, for inviting me for this lovely occasion of National Pharmacy Week in your international conference on natural and novel systems in the research and development, manufacturing, and other areas. So I really appreciate and your invitation, and it's always my pleasure to interact with the university students, professors at the various forums, in addition to the various conferences on regulatory, manufacturing, R&D, etc., etc. So today is my 66th webinar, and this webinar is special to me because first time I'm going to Jharkhand at Arka Jain University. So for me, it's an honor to be in the Jharkhand Tata Nagar area. And I was quite pleased when Dr. Sahu spoke to me a few weeks ago about this program. And we have some common friends. So I was really impressed by this activity by Dr. Sahu. And I was requested to speak by Dr. Sahu on the current scenario and future challenges in the product development. So I was discussing with Dr. Sahu on this subject, and where I told him that is it okay if we discuss quality by design in drug development or natural products? So he was quite pleased with this thought. And so now my presentation today is based on the quality by design concepts, current scenario, and future challenges, and what we all need to learn at college time during college activities, as well as when you enter the industry, what kind of expectations are there in the industry for the from the perspective of the this on today's subject. Now, my more focus will be on the drug development with few touches on the herbal products or phytosynthetic products, where I'll be discussing right from the beginning of identifying the process and how you can implement, why it is needed, first of all, why US FDA initiated this activity. So I would like to discuss that, and then slowly in the phase manner, we will go ahead with the our subject. So let me present my uh, presentation to you. You are able to see it?
Yes, sir.
Good.
It is visible.
Visible. So I'm changing the slide purpose and coming back. Tell me whether it is okay.
Yes, sir.
Good. Okay, so it's a generic drug development with quality by design concepts. Now, many a times people may be aware, may not be aware in the college career, the students, that quality by design concept, why it is required, and what are those rationales behind quality by design by regulatory agencies worldwide. And now, if you do not have your product development through quality by design, they may not accept your files for registrations. So common technical document which is needed by regulatory agencies now must have quality by design elements in the development process of the product. So keep in mind, my dear friends, students, that quality by design is must for highly regulated markets like United States, Europe, and uh, Australia, Japan. These are highly regulated markets. Even for that matter, PICS guideline also, they also are recommending strongly quality by design. And WHO also has started uh, appreciating a recommendation on quality by design. So let's see why this thing has happened and what kind of current scenario most of the companies have and who are in the process of changing to the new concept. Today, though this is going on for several years, several decades in the past, this kind of manufacturing activity with variables or the fluctuations in the manufacturing, though it is standardized, but there are always fluctuation due to the input materials and have a various variables or the parameters which are critical impacting the product which is coming out after the development, which is coming out after commercial batches are manufactured. And the main issue here is locked process variables, that everything is fixed and we have to comply within that range which is fixed. There, the flexibility is not there. And that was an issue that we didn't identify the factors affecting the variations in the process. And so that is why there was an impact on the quality of the product. And due to this fixed manufacturing process. And so why it happened then? What was the issue behind that? Is there were several complaints in the market, and those are due to lack of efficacies of the product. There used to be several side effects, and more than 60% issues were based on the quality compliances, and which was resulting into the several recalls from the market. Now you know what happens when there is a recall. When there is a recall, patients suffer. Products are not available in the market. Market is starving of those products which are useful to the patient and resulting into the drug shortages. So regulatory agency like US FDA decided to find out, do the survey, why these quality issues are coming up. And soon they realized that these quality issues are because of non-robust processes, which are not consistent in manufacturing. They tried to find out the reasons for that. Is it due to the haste to file the product, to register the product in competition, to achieve the target of filing? Whether company is taking any shortcuts to launch the product? Whether companies have sufficient resources? Are they having any low investments in development of the product by way of manpower, by way of machines, equipment, and the facilities? So this kind of approach they were thinking, and they definitely came to a conclusion. Now we have to intervene and form certain scenarios where we can introduce the system to ensure that no manufacturer will compromise on the quality for the development of the product. And that is why this whole concept of quality by design has come up, which here on your screen you will see, my friends, the comparison between traditional and the new system of product development, that is quality by design. And quality by design is literally, in the meaning, is designing the product and designing quality during the development of the process itself, ensuring there is a quality in the development itself, ensuring there is a quality of performance in research and development, which was missing in the past. And earlier, so the formulation development used to be trial and error with some data driven, but now it is more of a systematic multivariate experiments and with a prospective approach. So from fixed to flexible and adjustable within the design space. So what is design space? I will discuss with you subsequently. Earlier, it used to be fixed in process control. Now it is real-time monitoring of the critical parameters, critical processes during the manufacturing operations. Earlier, it used to be fixed, limited data of the batches, maybe monographs in the pharmacopoeia, or companies, whatever they have identified initially, fix standards. Now the things are overall control, not only the end product testing. Now there will be several critical processes will be monitored. And that's the big difference. So there is also an opportunity here to improve the process. Because in the past, it used to be once you file the document, once the document is released for manufacturing, it is standardized and hundreds of batches were getting manufactured. Though there is inconsistency in the outcome. Now, it is a continuous quality improvement throughout the shelf life of the product. So it is no more quality by testing. No more quality by testing. Now it's monitoring all critical steps during manufacturing. That is why it is called as quality by design. So you are designing quality into the product now. So what it involves? Right from patient's requirement. What patient wants? What kind of focus is required on product? Now, whole concept of quality by design is based on science and risk-based approach. Everybody starts thinking only on science, only on risk. So that also I will discuss what is risk and how it is to be handled. And this is a continuous improvement process throughout the life cycle of a product. And that is why the definition which has been coined by International Conference of the Harmonization or International Confederation of Harmonization. The definition, each word in this definition is highly loaded, my friends. So let's discuss this definition. Systematic approach, right? Now we are much better way developing the product and the R&D department in the research and development itself, which begins with predefined objectives and emphasizing on the product and process understanding. So we are focusing now more on product and process understanding during the development itself and ensure right process control strategy, which is based on sound science and quality risk. So overall, each word is highly loaded. And what are then these predefined objectives? These are the natural questions we may have. So predefined objectives are customer satisfaction. What kind of product customer wants and why? So let's discuss that in future slides. These are the key terminologies in quality by design concept. You will have to remember whole life. If you are going to enter in the pharmaceutical industry, or even though you start your own business. Quality target product profile, QTPP, critical quality attributes, that is CQA of raw material, active drug substances, supplies. Critical process parameters during the manufacture of the product or a raw material, drug substance. Critical material attribute, CMA, for finished product. DS, design space, for ranges of the critical parameters. And how will you control these ranges, ensuring they are within the defined norms as per design space is a control strategy. So remember these CS, DS, CMA, CPP, CQA, QTPP. So overall, if you see this whole concept of QBD roadmap, it starts with QTPP and ends with life cycle management of the product. So it starts right from patient's need on one side. Patient's need on one side for what on the target product profile. What target we have is a patient's requirement. And based on that, going towards the product development, process development, and to start commercial production. And from manufacturing side, we will have to ensure that from facility perspectives, process parameters, design space identification, the listing, the CQAs and the QTPP correlation with the help of risk management throughout, which is very critical. And therefore, QTPP is the first step in drug product development. Now, this drug product, or whether it is a natural product, whole activity remains the same, my friends. Whole activity remains the same. Whether it is natural product or drugs, activity is same. Nomenclatures are same. So this defines, QTPP defines safety, efficacy, quality, and manufacturability. So these are the quality characteristics that the drug product or natural product should possess in order to reproducibly deliver the therapeutic benefit to the patients, which is labeled by the company. That means label claim on the container or packs or the strips or the bottles. What the label says, so many milligram of aceto, let's say, we can say aspirin, for example, simple one, aspirin 500 milligram per tablet or 500 milligram per capsule. Are we giving that to the patient? They need 100% of the level playing. And therefore, from the safety, efficacy, and quality perspective, this is very, very important document of quality target product profile, which is helping to identify the goals and the risks involved during the manufacturing, stability, and the shelf life of the product. So there is a throughout customer focus, patient focus during the development itself. And the R&D department plays a key role here to ensure to identify right critical quality attributes, to identify the correct critical process parameters, generate the data to monitor it, and ensure that there will be a complete consistency after every batch during commercialization. And this is strongly recommended by US FDA, now Europe, now WHO as well. So one example I'm showing you of the drug product. Let's say take the example of immediate release acetyltryptan 20 milligram. So what this target product profile is, dosage form, dosage design is immediate release tablet. Dosage form is tablet. Route of administration, oral. The strength is 20 milligram. The pharmacokinetics, that is Tmax or bioequivalency in comparison to the patent product, stability of the product, a specification of the quality attributes, and the packaging system, etc., etc. So that comes under this kind of brief shown on your screen in the tabular form, which regulatory agency is always happy to receive. And we have to always give the justification for each of the elements. So this is how it should be represented even in natural product designing as well. This is the example of the herbal product. Look at here as a natural product. Some people call it as Ayurvedic. Some people call it as herbal. So let it be as it is. But important is the concept. The dosage form, tablet. This is for herbal product. Design, conventional release, tablet. Why? Patient's acceptability, easily they can swallow. Route of administration, oral. Dosage strength, 510 milligram for therapeutic benefits. Quality attributes are appearance, hardness, etc., etc., till friability to meet. This is exactly like drug product, my friends. Remember, natural products are not different than the drug product for quality by design compliances. So remember, the whole concept is also applicable to the natural products. Now, this particular example is herbal antioxidant tablet as a supplement. I'm sure you must be knowing in the herbal product extraction general procedures like solid liquid extractions, different steps, evaporation of the organic solvents if you are using, or aqueous if you are having a solid evaporation material coming out as a concentrate of the extract, then convert it to the granulation, and from granulation to the formulation, either tablet or capsule. So it depends how you look at this whole process for the development, depending on the need of the product, depending on the physical and chemical characteristics of the natural product as well. So natural product also has an impact of temperature, solvent, and flux during the manufacturing of the extract, remember. And this extract you are going to concentrate to be more effective product. Usually these extracts are critical in natural products. Now, these are some of the herbal excipients impact. Look at the excipients, some of the excipients required here, which are having an impact on disintegration of the tablet or friability of the tablet. And this chart is from one of the published articles on herbal antioxidant tablet, which I mentioned earlier as well, based on the software. And there are two factors, two variables. What are the variables we talked of? Is the variables having impact on the critical quality parameters. Now, which are the quality parameters? Disintegration. Now, disintegration has an impact on dissolution. So, what is important is if you address correct excipient proportion in the tablet, your dissolution will definitely comply. That is how you have to trace back where it can fail. And if you feel that starch is critical, you have to plan your experiments. The combination of starch and CCS also plays an important role. And this is what has been displayed here in these 9 experiments of design space, and which gives you that which is the right combination which you need for a better disintegration time, and then which has a direct impact on the dissolution profile of the tablet. So it is whole activity of quality by design is transition to reduce the variability in supplies in manufacturing and to ensure the consistency in the real-time quality assurance. So now the new concept is the raw material perfectly designed into the manufacturing process by identifying the critical quality attributes and critical process parameter, and have a right product at the end, and continuously give this feedback to R&D to ensure the product is improved throughout the shelf life. Now look one example here. Now we talked earlier of antioxidant tablet, right? In herbals, similarly, this is a drug product. Now here, both antioxidant tablet of herbal and drug product is same. Now, we talked of what is important is initially to develop concept of quality by design is patience. Now, if you are having a running nose and having a severe cough and cold, what do you need? Quick relief. What patient wants? Quick relief, because there is a headache and continuous nose is running. So what is important that we should give the product? Quick relief to the product. Now quick relief can come when the tablet has a quick dissolution, and the quick dissolution will come when hardness is complying. Quick dissolution will come when disintegration is perfect, and also there is a relationship with content uniformity of active drug in each tablet. So what is critical to the patient is dissolution. What is critical to the patient? Content uniformity, and which is impacted by hardness, disintegration, and excipients composition. So we have to address excipient composition to get proper dissolution, to get quick relief to the patient. That is how the whole process steps. CQA identification and CPP. CQA is critical quality attributes. CPP is process parameter, critical process parameter. So during mixing of the product, ingredients and excipients, what is important? CQA is potency. And what is the process parameter important is mixing time and mixing speed. One example, look at this. So how do you monitor and control the whole mixing time? And the speed of mixing is critical for a right potency, right CQA. Though CPP affecting the quality attributes, therefore it becomes CQA. And so during the mixing, potency is most critical attribute, and which is affected by mixing time and mixing speed. This is just a small example. Same way, in the depyrogenation sterile product, pyrogen free we want as a quality attribute, which depends on proper sterility generation, proper depyrogenation of the packaging material or the area which is related to the temperature. So how do you monitor the speed of the vials running through the bottles running through the tunnel after washing and drying, and at a highest temperature, how it is monitored to make it pyrogen free, and the temperature inside the tunnel. So this is how we have to identify. So everywhere risk assessment is required during the development of the product and during the manufacturing of the product. So this risk assessment can be identified, the risk at each step of development manufacturing. There are different tools. You can have a qualitative risk assessment, that is like road signals, red, yellow, and green, which you see on the right side of your screen. Initially, it is cause and effect analysis, 6M. You must be knowing fishbone concept. And quantitatively, you can do FMEA technology or FMEA concept, which is giving ranking. So from that perspective, this kind of risk assessment to be identified quantitatively or qualitatively, both are okay, no problem. In case of herbal preparation, this is one example I'm displaying here for ethanol preparation. What can go wrong in ethanol precipitation? So recovery of API and removal of saccharides in one of the example which is there in the publication. And they said that what concentration of ethanol, whether anything can go wrong in ethanol addition, anything can go wrong in concentrate of the extract, whether it is pH or a temperature or a density, same way in refrigeration equipment, environment, etc., etc. So how do you represent it? So right from drug product CQA vis-a-vis the parameter, process parameter, like in some cases, it may be identification can be serious in API because it is a critical. If the own original API is not correct, what's the use of using in product? So identification is very important of the product. Assay, content uniformity, impurities, dissolution, these are all critical from API perspective. During the processes during manufacturing, we relate this criticality to the dissolution and blending. So we have to identify the critical process parameters vis-a-vis critical quality attributes impacting the product quality, impacting the patient. So that's how whole relationship happens. So our whole monitoring and generating the process monitoring activity, generating the identification of the correct critical process parameter and correct right critical monitoring of critical process parameter will help you to generate, remove all the criticality and bring whole scenario into the green representation. So this is where you are nullifying the effect of the seriousness or criticality of the process parameter. So when you do the design of experiments, even in the, now this is one example of a herbal product where density concentrate in grams per mil at temperature 20 degrees or ethanol consumption required or setting temperature. Some variables are identified here for running various experiments for design space. Design space, in other words, is nothing but identify the specification ranges between which you have to identify specific normal operating range for daily operations. And this is going to be helpful for us in monitoring the process. So when you identify the design space, we have to ensure my parameters are within my process parameter, critical process parameter monitoring are within these ranges identified based on design of experiments and generating design space. Another example of design space, how you can see through, is this is again a herbal product, design space identification for ethanol consumption. What kind of ethanol, how much percentage you need for the extraction of the product, or rather for the extract of the herbal product, and the concentrate required, what thickness, what concentrate percentage you need. And then this is a contour plots for 3D. You can see on your software. You don't have to worry much about this. Your software will tell you what to do, and software will give you on the screen all the data. What is important is identifying the right design space and feed right information to get this kind of output, and this will guide you correctly what should be my design space for ethanol consumption vis-a-vis the density concentrate. So there are several great opportunities for quality by design for us. Is increased process efficiency, uh, the higher level of assurance for the drug product, transparent and predictable outcome. So you can know much before your final steps happen, the whether are you moving in the right direction to get the quality product. So this whole process is also known as quality predictable process, and very transparent. Everything is fully automatically uh, documented. Remember this. Nobody can tamper with it during the commercial batches manufacturing, and it is minimizing the quality issues, compliance issues, and you save a lot of millions of dollars on the penalties coming from regulatory agencies. So it speeds up as well for the manufacturing changes and the regulatory filings for any changes, and you are being moving towards continuous improvement of the product throughout the shelf life. So you are moving towards ensuring patient safety for developing the product by way of increasing the consistency batch after the batch, unit after unit in manufacturing. So design space is critical, and robust product and process development will always help you for transparent operation. Nobody can compromise on that, and organization uh, is going to achieve right first time in development, right first time in manufacturing as well, because now you are giving to manufacturing robust process for the during the manufacturing commercial batches, and therefore we are moving towards zero defect now, zero defect in commercial manufacturing for better productivity and process predictability. So whole concept is revolving around science and risk-based understanding during the development itself, by identifying right critical quality attributes, right critical process parameter, and the impact of interaction between the excipients and the active drugs. There is another concept also in the quality by design we talk of. This is also nowadays in certain part of Europe, the process analytical technology is being used. Some people have some apprehensions here, which I'm going to talk to you. They are worried about the cost, but it is one-time cost, but it's a longer time benefit. Remember this, because your accuracy of monitoring critical process parameter and moving towards the next step increases your productivity by almost 100%. And this is a reality. We have experienced it. So you can have, there are four types of process analytical technology, where we talk of inline, that is a sensor is in connection with the product. Now that concept is changed. Without touching the product, they can sense the activity of the drug there, the optimum conditions of the drug, and automatically it is analyzed. You get the printed data. You don't have to sample at all. Same thing inline and online. But at line, in the production area, you draw the sample out and analyze near your production area. or offline you send the sample to quality control. So the process analytical technology is, my friends, not only analysis. Remember, it monitors your critical processes and identifies the, if anything is wrong, immediately there is a message across the key people, the way in which you plan your system, and reaction stops there. It doesn't move forward. So you have a time to make correction rather than rejecting and destruction of the batch. So you have tremendous cost saving. This also we have done ourselves, experienced, and by using this, we have saved more than $70,000 per annum. So it's a great tool if you spend initially money on these capex. One example for drugs from the drug perspective, which I have picked up this slide from US FDA presentation, and the similar concept is also for natural product existing today. There are several publications on this as well. So right from dispensing to the coating of the tablet, every critical steps you are monitoring by various tools, which are called real-time release testing. And this real-time release testing is nothing but process analytical technology, monitoring all critical process parameters by way of analytical tool. So monitoring and analysis of process parameters simultaneously. But there are, of course, when you go for so much high automation, there were challenges in the organization, my friends. The biggest challenge is cultural, that is a quality culture. Right from top to the bottom, everybody have to ensure, but topmost person of the company must ensure he himself follows the quality culture and doesn't pressurize any quality team for releasing the substandard batches. Knowledge, yes, knowledge is the key. Training is the key. Avoid any pressures and speed. This is for the top management. For the R&D team, it is a risk understanding and remediation of the risk. Avoid resistance to implement the new technology. Cost is not an issue. It is a one-time cost. It is always there initially. But when you look at the long term, and I give you assurance that if you implement this concept, your return on investment is maximum 3 years, not beyond that. There are certain challenges even in development of the phytopharmaceutical drugs, and some of the challenges, no doubt, of course, I had one of the important thing is need for investment, which is there everywhere. Need for, without that you can't get returns. But here some people have an issue of lack of regulatory experiences for this kind of category of the drug. So some of the guidelines are also vague. Some guidelines are not there which are required. Now, just for your information, my friends, regulatory guidelines are always there for everything, but they never tell how to implement it. Is the privilege of the manufacturer or the company. Now, in case of herbal or natural products or phytopharmaceuticals, most critical is the right quality of the input raw material. Active raw material, the source of that plays an important role. The soil plays an important role. Keep in mind that that kind of things give you a different perspective of your data. Another challenge is pharmacovigilance, and the whatever data you generate by way of clinical trial is today insufficiently documented. It's a very big challenge the natural product industry has. There is many of them are copy paste and manufacture the product, market it. If you ask them the data on clinical trial, they will have a big question mark. Insufficient data on clinical trials in natural products is also a big challenge. Then how do we move forward? Fundamental shift in our thinking. Yes, quality culture, operational excellence are the two key founders for achieving the quality by design. in drug product as well as natural product as well, which is same, no difference at all. So the new paradigm, risk-based approach in natural product development. The risk is that, am I assuring my patients that clinically they will be, my product will be useful? Do I have that data I have to generate? That's a risk if I don't generate. And you will be embarrassed if the nowadays lot of awareness is generated in the market about clinical studies of the natural products. And many people are not able to answer, many organizations, on this. So it's a big risk to have a business from the perspective of clinical trials. So quality system, of course, same, natural or drug. So you can use these guidelines for natural products as well, though they don't bring it into the scope by regulatory agencies. But this will be a good guideline for natural products as well, in addition to the drugs manufacturing. So it is a whole thing. We are now moving from blind compliance towards science and risk-based approach. From current state of quality by design to the desired state, it may take more than a year or 2 years for implementation, fully understanding, training, and generating and ensuring that everyone in the organization gets this message. The cascading down the information on this and training will help tremendously. And to achieve when you start quality by design, you are reaching towards 6 Sigma compliance. Remember, this is not a joke. Today, in drug pharma, we are in the 2 to 3 Sigma compliance. I cannot comment about natural products, but certainly the drug industry is moving towards 6 Sigma compliance. That is for sure. I would love to see natural product industry coming to this level of 6 Sigma by ensuring clinical trial, which is very critical. So the initial cost, when you achieve the desired result, when QBD is fully realized, your cost goes down. Remember, rejection goes down, reprocessing goes down. That itself recovers a lot of cost every month, every year. Future challenges. Now you look at these automations, what are going on today. The software, world of software, telemedicines, everybody knows now, e-medicine, artificial intelligence, Internet of Things, supply chain automation, blockchain, and these are going to play a major role in time to come. We have to educate ourselves. We have to be literate on digital technologies, my friend. Ensure that you start learning. There are several free courses available on these. Whenever you need that information, please let me know. I can guide you how to access the free education on digital literacy for the students and the teaching staff, as well as for the industry as well. So there are so many potential use of artificial intelligence in healthcare industry, which includes natural product as well. And right from throughout, from quality, supply chain, manufacturing, R&D, AI and robotics, people have started using it since last 4 years. In India, we have started using this technology for restaurants for delivering the dishes on the table. In some of the restaurants in Bangalore and Hyderabad, they are using this. So why we adopt these newer technologies? Robust process, reducing risk to the patient by way improving the patient benefits, reducing the cost. Of course, it reduces the cost. By way of minimizing the variability in the batch after batch, unit after unit, and shortening the time to launch, improve the credibility of the organization. Thank you, my friends. I could, whatever I could do the justice for the time given, 45 minutes to me, which we started at 10:20. And I think I have finished in 45 minutes as requested by Dr. Sahu. And I hope you have understood the concept of the whole development perspective for natural product as well as for the drug product. Thank you very much, Dr. Sahu, once again for the invitation and giving me this opportunity for your university. Thanks a lot.
Thank you, sir. Thank you for this wonderful session. We are very privileged to have you, sir. Now, may I request to our Dean, sir, Dr. Jyotirmay Sahu, to kindly felicitate Ranjit, sir, with small token of appreciation.
Thank you, Kusuman. Just a minute, please. Just a minute. Because some problem is going on.
Abhishek?
Yeah. My video is not getting.
Yes, sir, you are not visible.
It's okay, it happens. Don't worry, sir. Technology definitely has some
Abhishek?
negatives.
I'm just going to you to this another gadget. I'm sorry for the things. Just a minute. Thank you, sir. I think is it audible?
Yes,
Yes, sir.
I'm seeing the screen as well.
I just want to speak something before the formalities. I just want to speak something. There are so many people attending this international e-conference, even from Philippines and from India, and from Indonesia, from Pakistan, and a few other countries. I am my whole heart gratitude to all of you to make this event wonderful only due to all of your support and contribution, and especially Dr. Ranjit sir. We are fortunate to have you with this e-conference as one of the potential speakers, sir. And whatever I have with me, that is not exactly to honor you. It is insufficient, I know. But still, though being an Indian, I like to give something today. So, Abhishek, my culture speaks that one. Abhishek, please, a small token of appreciation on behalf of San Pedro College of Philippines and Arkajan from Jharkhand. Please accept, sir. Definitely, I have get a lot of information regarding QBD from you. It is a part of our syllabus today for the pharma students. But the teachers are not, and me also being the professor from CUTI's background, not that much uh, educated with this one. Because might be in my time it is not in our syllabus, and we people have also not focused a lot on that part due to our lack of interest might be. Because if you focus and try to culture on this part, then definitely we must have time to uh, develop this knowledge. But regarding QBT, I able to get know in the year 2016 from one of my friend who is working with Origin Pharma as the plant head, Dr. Vikas Naik. He has done his PhD on this one, QBT. So he educated me. He is one of the advisory committee of this international conference. He educated me what is QBT and what is the objectives of this one. and gradually some things getting cleared in my ideas what is QBT and why it should be done. But particularly in academic sector, it is not yet full-fledgedly applied. So with your help and in upcoming days, even I will take few classes, sir, with your cooperation. पहले मैं पढ़ूंगा बच्चों को पढ़ाऊंगा. Thank you, sir. Thank you for your kind and benign presence and for your information.
Can I come?
Yes, sir.
Can I come?
Yes, yes. Please tell, sir.
See, you, anytime you need any kind of help, when I'm saying any kind means any kind, I'm always there. And once this pandemic gets over and we are sure that it has improved, I will definitely come there. I will definitely take several sessions on quality by design for your professors as well. Because professors having literature in this area, because this kind of area is the tomorrow's pharma industry, tomorrow natural product industry. And it is required to comply with that if you are interested to export the products to a highly regulated market for a better business. So this is a key, and this will always give you tremendous cost benefit. In natural product area, people are not realizing this concept. And whereas even in drug product, very few companies have started implementing, the top 25 or 50 companies, not all. In India, there are more than 10,000 companies. And almost 700 companies are exporting the products to US and Europe, highly regulated market. So from that perspective, the drug industry also needs to pick up this concept. And without these, you cannot file the applications to US FDA or Europe now. So this becomes a key thing for students to understand. And when they enter the industry, they go well prepared, because we see a lot of differences between the students, the curriculum of the universities vis-a-vis the industry requirement. There is a big gap today. And I am doing a lot of sessions with several universities and the industries as well, how we can fill this gap, what should be done jointly. So I'm talking to the communicating with your PCI chairman also on this for last more than a year.
Yes.
So I'm doing my best, but I'm always available at your service, doctor.
Thank you, sir. You are wonderful words. I like to introduce you even, sir, to the director of Philippines San Pedro, Dr. Erwin, and the dean, Pedro. And our next plenary speaker is Dr. Nagaraj Rao, who helped me a lot. He is also my MOU partner, and I am really grateful to these people. We are very new school, sir. School of only 3 years. It is yet, in my language, the school is under the soil. It is not yet germinated. So it only in 6 year, you people like the blessings of you people, cooperation of you people in that, by the virtue of that pitch, please, sir. Thank you. Thank you. Sir.
Bye for now. Bye.
Thank you, sir. Now, moving for our next session, we have Dr. Nagaraj Rao, Managing Director, Rane Rao Reshmiya Laboratories Private Limited, New Mumbai, India. Good morning, sir.
Hey, good morning.
Good morning, sir.
After a long day.
Hello.
After a long day, I am getting you, sir, virtually, but I feel I am physically seeing you, sir. And I am feeling excited, sir.
My pleasure. Thank you.
Please,
Sir has
please,
obtained his yes, sir. Sir has obtained his bachelor degree in science, chemistry, and in technology of pharmaceutical and fine chemicals from the University of Mumbai. After working with Colgate Palmolive India for 2 years as a laboratory chemist, he obtained his doctorate in science from a reputed university of Germany and also carried out his postdoctoral research at the Institute of Biotechnology of the Research Center Jülich, Germany. He was a member of the editorial board of the Office German language version of the first European pharmacopoeia. He was a visiting scientist at Jülich and a visiting faculty of the Institute of Chemical Technology, Mumbai from 1993 to 2007 in the field of bioprocess technology. He has authored several original research articles, a patent, review articles, and book chapters in the field of pharmaceutical biotechnology, brewery, and surface coating. Welcome, sir, in this virtual platform of International E-Conference. We are overwhelmed to have you for today's plenary session. Now, may I request you, sir, please kindly enlighten the session with your valuable knowledge. Over to you, sir.
Yes, thank you very much. Is it possible to display the PPT which I had sent yesterday?
Yes, sir.
Please
Time.
share
It
the
will take
PPT
little
on the
time, sir.
screen.
Abhishek, please share the PPT.
Yes.
Khushbu ma'am, it will just take
Meanwhile,
time.
I would like
2 to 3 minutes.
to.
2 to 3 minutes.
Okay, sir.
Yeah.
Okay.
I would like to take the chance till the PPT is uploaded to express my admiration and gratitude to the Arka Jain University for being in touch with us and interacting with us, and also specifically giving me here today the chance to speak about the subject which is very close to my heart. Since this international conference is being organized by the schools of pharmacy of San Pedro in the Philippines and Arka Jain University, my talk will be focused on what the schools of pharmacy can do, or where the opportunities lie hidden for the students when they come out of the pharmacy colleges, all in the backdrop of the development of vaccines in India, specifically in the last 1.5 to 2 years. My talk won't be very highly technical. It will be more trying to highlight how vaccine manufacturing is closely related to many of the subjects which we study during our pharmacy courses. Just now we have heard about quality by design that is necessary everywhere, and so once again, thanks to the organizers. It is very difficult for me to say no to Dr. Jyotirmay Sahu whenever he requests something. And let us get on with the presentation here on novelty in the vaccines which are being developed or have been developed in India. May we go to the next slide? Yes, this gives you an idea of how many vaccine candidates are being in clinical trials across the world as per the World Health Organization statistics. These statistics are updated almost every day, and these figures are from a few days back. This gives you an idea that different types of vaccines are being tried in order to overwhelm the coronavirus, the COVID-19 virus, and try to give protection to human beings from either getting infected, or if they get infected, to survive and get healthy. Protein-based viruses dominate the field. There are 47 of them. Viral vector-based non-replicating types are 19 of them. Inactivated viruses, you have 17. Messenger RNA-based viruses are 21. And DNA-based viruses are 15 among the total of 132 where clinical trials are in progress. And interestingly, in all these 5 categories of vaccines, we have Indian companies who have developed the vaccines for all these 5 categories. Can we go to the next slide? We will begin with the vaccines developed by Bharat Biotech, because Bharat Biotech is, for all types of reasons, always in the news. And the example of Bharat Biotech symbolizes many things. The vaccine, which has been allowed now for emergency use by the World Health Organization, was named BBV152 by Bharat Biotech. It is a whole virion vaccine, means it is an inactivated vaccine with an outer protein shell and, of course, the inside nucleic acid core. And each of you who has taken Covaxin has got 6 milligrams of this vaccine in the 0.5 ml, which was injected into your arm by a nurse or a health worker. The pH of this solution in which the protein is suspended or dissolved is fairly around neutral, around 7 to 8. The vaccine, the virus as such, is extremely dangerous, and I mean, if it spreads out of contained equipment or laboratories, it can cause havoc. That was the challenge which Bharat Biotech was facing in February, March 2020, when everybody, all the authorities, whether in the US or China, accepted that yes, there is a pandemic which is happening, and we don't have proper tools to fight it. At that point of time, the National Institute of Virology, a branch of the Indian Council of Medical Research, was able to isolate the virus from a person who had just come into India from England. They could do the genetic mapping, and then they could isolate the virus and characterize it. And the National Institute of Virology announced this and invited companies in India to come forward to use this virus in order to prepare a vaccine. Bharat Biotech took this request seriously. Probably it was the only company which could, at that point of time, take it seriously, because these viruses can be handled, only in very special types of safety laboratories, which are called the biosafety laboratories. Have different levels of safety: biosafety level 1, level 2, level 3, level 4. And these viruses can be handled only in biosafety level 3 and 4 laboratories. Bharat Biotech, being the only company in India having a BSL 3 facility, took up the challenge. And if you have been following the media for about 4 months, all the biotechnologists, the microbiologists, the analytical staff, all huddled together in apartments very close to the Hyderabad company of Bharat Biotech. And it took about 4 months for them to come out with the vaccine. When the virus is very toxic and potent, pathogenic, it is inactivated by using certain organic chemicals. And in the case of Bharat Biotech, they used beta-propiolactone in order to inactivate the virus. Beta-propiolactone is imported in large quantities, gives you an opportunity to think of manufacturing in India. Now, the technology of using inactivated viruses as vaccines is known since a long time. I mean, we have done remarkable progress with the polio vaccine, for example, which contains inactivated viruses. What happens is that it is known that the activity of the inactivated virus can be enhanced by adding certain types of adjuvants. And these adjuvants are added not only to inactivated viruses, but also for mRNA types of vaccines. Also, they are being investigated. What Bharat Biotech did was to use an adjuvant called imidazoquinoline adsorbed onto alumina gel. And this imidazoquinoline is called as a toll-like receptor 7/8. By having this combination of the vaccine of the inactivated virus and this adjuvant, the potency of the vaccine was remarkably enhanced. And so on the one hand, you have a classical technology being successfully tried and implemented. And on the other hand, you have the experience with adjuvants being positively used to enhance the efficacy of this vaccine. Let's go to the next slide. Continuing with this co-vaccine, one thing which needs to be highlighted is that parallel to the requirement of Bharat Biotech, when it realized that it needs imidazoquinoline in large quantities, the Indian Institute of Chemical Technology, one of the laboratories of the Council of Scientific and Industrial Research, came up very fast with the manufacturing process for imidazoquinoline and also method validation process for the same. So you can see how a research laboratory collaborated with the industry in meeting its needs for a raw material. Once the once the company had got the virus from another national laboratory and prepared the inactivated vaccines. This virus strain is called NIV 2020-770, NIV for the National Institute of Virology. And it contains aspartame 614 glycine mutation in the spike protein. And as I said, it was isolated from a COVID-19 patient flying in from England, and it was sequenced at the National Institute of Virology. Next slide, please. As I said, Bharat Biotech had a BSL-3 laboratory where they could work with the highly toxic virus. Over a period of time, looking at the lack of BSL-3 laboratories in India, the Indian government has been very proactive, and today at different locations in India, BSL-3 laboratories are being set up by companies at very high speed, breakneck pace, and the first industrial BSL-3 laboratory to produce Covaxin and others which require such safety facilities will be starting producing in Gujarat in February or March next year. In Mumbai, Hafkin Institute is in the process of setting up the BSL-3 facility, and there are others also who are doing that. Among the other things which got simultaneously developed was, fortunately, the Covaxin of Bharat Biotech could be saved, stored, transported at 2 to 8 degrees centigrade. I mean, if you compare that with the Pfizer-BioNTech vaccine, which has to be stored at much, much colder temperatures and transported, the entire logistics industry in India geared up to the crisis, and today it is very easy to for temperature control logistics to be done for vaccines, medicines, and whatever which requires cold storage during transit and during storage. So that is one offshoot of the Covaxin which was developed, that temperature cold storage facilities and logistics improved dramatically. We'll come to some more other side effects, positive side effects as we move along. Another Indian company has been doing research in-house at their biotech research center in Ahmedabad, Zydus Cadila. They are one of the very few companies who have dared to try a DNA-based platform, a plasmid DNA-based platform to develop a vaccine against the COVID-19 virus. And the vaccine which has been developed by Zydus Cadila, ZyCoV-D as it is called, has about 67% efficiency in the first clinical trials which have taken place. And fortunately, it can also be stored at 2 to 8 degrees centigrade. Probably what attracted Zydus Cadila to go for the DNA platform is that when it comes to comparing the different technologies to manufacture vaccines, the manufacturing of inactivated whole virion vaccines is a very slow process and a tedious process. Compared to that, adenovector viruses are a little faster to produce, and mRNA and dRNA vaccines are much, much easier to produce, which means you can scale up much more easily in order to meet the demands. Probably that was one of the reasons which attracted Zydus Cadila not to go via the mRNA route, but directly to jump into the plasmid DNA platform. Fortunately, the initial clinical trials have shown that the vaccine is efficient and the side effects are within the tolerances set by the authorities. One interesting thing about this Zydus Cadila vaccine, what is important for our students studying pharmaceutical technology, is that it is a subcutaneous vaccine, unlike most of the vaccines which are intramuscular. So the ZyCoV-D vaccine uses a needle-free intradermal applicator in order to get the vaccine under the skin and have its effect. All the Indian companies have been working closely with the laboratories, with the government laboratories and universities, and they have been also funded well by different organizations within the government. Here is an example of the Department of Biotechnology supporting Zydus in the development of its vaccine. So this is an example of a complete Indian R&D leading to a successful development of a vaccine. Next slide, please. Yes, a little more about the way of administration of this Aedes aegypti vaccine. The needle-free injector is used. This involves an applicator which delivers the liquid through the skin to the proper layer of the tissue. So developing small gadgets, small devices, administer such vaccines is something which in pharmaceutical technology there are quite a few opportunities. Next slide, please. Now we come to another vaccine which is developed by a relatively smaller company called Genova in Pune. It is the first mRNA, messenger RNA based vaccine which has been developed in India in collaboration with HDT Biotech Corporation in the USA. It is a self-amplifying mRNA vaccine, which means that once it enters the inner core of the virus, it starts multiplying by itself and then leads to the death of the virus. One important feature of most of the mRNA vaccines is that they are destroyed very easily by the enzymes within the body. So we need a delivery mechanism which protects this mRNA vaccine until it reaches the target. And this has been achieved by what is called as a lipid inorganic nanoparticle emulsion technology. It's called LION technology. And this lipid-based nanoparticle emulsion protects the protein from getting hydrolyzed by the enzymes until it reaches its destination. This lipid-based nanoparticle emulsion technology is another challenge which our pharmacy colleges can take up to develop more and more alternatives to the first one which has been put in the market. So this vaccine is called HGC019 and has all the information to guide the host cells to make the antigen spike protein of the virus, reported to interact with host cells receptor, and is supported by this lipid inorganic nanoparticle as a delivery vehicle. This is a technology which has been patented, but as you will see, there are many options possible once you know that an outer lipid shell is able to protect the inner protein. Next slide, please. Yeah. Most of the mRNA vaccines which are encapsulated use the encapsulation technology with the lipid membrane, but the Genova scientists have used an adsorption technology in order to attach the mRNA to the lipid carriers, nanolipid carrier surface in order to boost the release kinetics of the mRNA within the cells, because adsorption forces are rather weak compared to when the protein is encapsulated, and therefore the release is relatively faster. Yeah, the development of this lipid nanoparticle delivery vehicles began originally by the collaboration of Moderna with Lonza. And Lonza is today the market leader, technology leader in this field. But as I said, there is still a lot of scope in that. And the key raw materials which are needed for these lipid nanoparticle delivery vehicles are manufactured by Evonik, and Evonik is setting up huge capacities in Germany near Düsseldorf in order to meet the demand for such raw materials for making the lipid shell for the mRNA vaccines. So you can have ionizable, cationic, and sweeter ionic complexing lipids, so that opens up a new field to do R&D and come up with new solutions for delivering the mRNA-based vaccines. Yeah. Now, one thing here, what has to be mentioned is that the budget, the expenses which Genova invested, and also the support which it got from DBT BIRAC, it was in the scale of a few crores of rupees, maybe about 15 crores of rupees. And normally, such research is possible by large corporations when the budget is at least 200 to 220 crores of rupees. And of course, the time taken by Genova has also been very less. So this is a tribute to the scientists at Genova that they could develop such a vaccine at very low cost and very rapidly, using an innovative technology of adsorption rather than nanoencapsulation, in cooperation with HDT of Seattle, USA. Next slide, please. Yes, Serum Institute is India's largest manufacturer of vaccines. They have very huge capacities because they are a provider to many countries across the globe of many of the vaccines which are life-saving vaccines, which we all know about. Serum Institute, as you know, could foresee that, there will be a big demand for a vaccine against COVID, and therefore, when Oxford and AstraZeneca announced that they are looking for partners who would manufacture the vaccine, Serum Institute moved in very fast, and the dedicated facility in Pune was set up to manufacture the vaccine, which is sold in India under the name of Covishield. This Covishield vaccine is a modified chimpanzee adenovirus CHAdOX1, as it is called, vaccine. That adenovirus protein is used as the vector. Now, it might appear very easy and to take for granted that when Serum Institute is working with Oxford and AstraZeneca, I mean, all the technology comes from them, all the protocols come from them, so it's very easy to replicate and start. But in reality, it is not like that. One has to adjust to the local conditions. One has to do the practical experiments and confirm what has been observed in the laboratories of AstraZeneca. Just getting a know-how and trying to produce, it does not happen that easily. It involves many steps, many trials, and maybe sometimes even a few disappointments. Serum Institute also is the dominant player in the Indian market for vaccines, as well as worldwide, as I said. And together, all the Indian manufacturers cater to more than 80% of the global requirements when it comes to numbers. So Indian companies, for every 100 people in the world who are vaccinated, at least 80 of them have been vaccinated with a vaccine developed in India. That is from the numbers point of view, but unfortunately, when you look from the value point of view, it is just the opposite. The 20% numbers which are sold by the other companies across the world, there are only 5 or 6 of them. They take away the cream with about 80% of the value. This was the scenario till the pandemic broke out, and I'm sure that this ratio is going to change because Indian companies have shown that they can do research and develop vaccines on their own, because facilities for manufacturing and logistics are coming up, and because we have very reliable and dependable clinical trial results, organizations which are doing that, doing a tremendous job. So instead of being a vaccine manufacturer only for poor countries, India is gradually emerging as a player to manufacture high value vaccines for the regulated markets. And this will be a positive development in the coming years. Just like Bharat Biotech 0.5 ml, you had 6 micrograms of the protein. If you got injected with Covishield, then there is a printing mistake here. It's 5 into 10 raised to 10 viral particles, which you got when you got the first dose of Covishield. Recombinant replication deficient chimpanzee adenovirus vector encoding the SARS-CoV-2 spike S glycoprotein. It is produced in genetically modified human embryonic kidney HEK293 cells, and the product contains genetically modified microorganisms. Next slide, please. Yes, Serum Institute naturally is working on several other newer vaccines, and one of them is with 2 other partners, Exelagen and Spy Biotech. Technology is for virus-like particle vaccine. Next slide, please. Yeah, this vaccine is called a hepatitis B surface antigen virus-like particle vaccine, and phase 1 and 2 clinical trials are going on. Likewise, Serum Institute is collaborating with Codagenics to manufacture live attenuated virus for nasal single dose application. This is branded as Covivac, and it's a single dose nasal live, intranasal live attenuated vaccine generated using Codagenics' proprietary de-optimization technology. Unlike many other COVID vaccines, this vaccine is designed to produce immunity against all SARS-CoV-2 proteins. See, the vaccine Covaxin has similar properties. For different variants, different mutants, it responds, it is effective. Whereas when you come to mRNA and DNA vaccines, because they are specifically designed against specific spike proteins, one has to check whether they're effective, or you have to tweak them in order to fight the new mutants. Not just the spike surface protein, this protects the patient against a range of SARS-CoV-2 strains. Live attenuated vaccines like Covivac and Covishield elicit a robust immune response and are associated with long-lasting cellular immunity. This is a known fact here. Next slide, please. Biologically, in Hyderabad is also a traditional and dominant player in the vaccines market, and their Corbevax vaccine has been recognized, been allowed, permitted to undergo phase 2, phase 3 clinical trials in India. It's also adjuvanted. It's a protein subunit RBD receptor binding domain vaccine. Next slide, please. Biologically, Evans, as earlier called, works with a large number of institutes across the globe. We will come to that later. Now, Indian Immunologicals Limited is a public sector company located very close to Bharat Biotech. And basically, they are competitors. But when it came to the manufacturing of Covaxin, and when the capacities had to be ramped up, Bharat Biotech and Indian Immunologicals collaborated with each other, exchanged the know-how, the technology, scientists, and today Indian Immunologicals manufactures the critical substances, raw materials required for Bharat Biotech in a very positive development, you can say. Instead of competition, there is cooperation. Indian Immunologicals naturally also works with other institutes across the world, and with Griffiths University, they are trying to develop another live attenuated virus. The other public sector companies which are planning to make Covaxin are Haskell Institute and BIPCOL, and in the private sector in Gujarat, where a cluster for manufacturing vaccines is coming up, Hester Biosciences. Next slide, please. Sputnik vaccines, which were originally developed by the Gamaleya Institute, technology transfer is taking place to Indian companies, spearheaded by Dr. Reddy's laboratories, and the others who are working on it, scaling up the technology, include Panacea Biotech in Delhi and Himachal Pradesh, Shilpa Biotech in Karnataka, Stelis in Karnataka, and Vichao in Telangana. Next slide, please. Yes, here I want to focus on the aspect that in such a complex field as vaccine development and manufacturing, it is not possible to do everything alone. There has to be collaborations, and there has to be a vigorous exchange of ideas, knowledge, technology in order to reach the goal fast and in a healthy manner. In the next slides, you will see examples of which companies are collaborating with which institutes or organizations. You can see Bharat Biotech collaborating with several government institutes in India as well as in the United States, and also several universities in the United States. Next slide, please. Serum Institute, again, a whole range of universities and companies with which they are collaborating across the globe. Next slide, please. Biologically, as I said, also is working with many companies in USA, Europe, and Japan. Next slide. And Genova also is working together with reputed institutes, not only for the vaccine against COVID-19, but also for developing technologies for potential use when new viruses crop up in the course of time. Next slide. Probably this is the... Yeah, and Zydus, of course, also with companies and institutes across the world. It is because of these partnerships that things happen faster. But there are two aspects to it. One is the partnerships enable you to develop the technology and also use the technology for scaling up. And then the question comes when scaling up has to take place. And there you sometimes find bottlenecks when it comes to fermenters, when it comes to, anything and everything you need for mammalian cell culture, for fermentation, for downstream processing, for analytics. So that is another field where in India, more emphasis is being given to become more self-reliant. Yes, some of the factors which have contributed to the success of the vaccine manufacturing, the vaccines programs in India are listed here. I forgot to mention or list here the clinical trial, the people associated with clinical trials. Those are the volunteers who dare to offer themselves to unknown vaccines. Then the frontline workers, the healthcare people. We are fortunate to have a active government, which is trying to make and has been fairly out of the box. Their first time in the history of government has been paying in advance to companies for products they will buy. So that has helped a lot. Drug registrations like the CDSCO and the technical committees, they have been very practical, they have been very pragmatic, they have been very fast. All has contributed in a positive and negative way. The overall free democracy, they have also played their part. I mean, one of the careers pharmacists can pursue later on is in journalism, health journalism. And because one could see that many of the reports which came in the media, they did not have a scientific basis, or people were writing reports without understanding what it was all about. Yeah. So that is one field where students can later think of going if they like journalism. So these are some of the reasons which have helped to contain the pandemic to the best extent possible. Salute to all the frontline workers, all the people who have made that, and thank you for your attention.
Thank you, sir. Thank you so much. This was really an informative session, as we get to know about different vaccines and its characteristics. Thank you. Um, Now, may I request to Babita ma'am, Dr. Babita Sarangi, Associate Professor, School of Pharmacy, Arkajan University, to kindly felicitate Sir. Abhishek.
We say
Hello.
Yes, ma'am. You are audible.
Good afternoon, everyone. Thank you, sir. I extend my gratitude to our distinguished speaker to take out time from his busy schedule and to grace the event. Thank you so much, sir, for sharing your knowledge and experiences. Your presence have made this e-conference very meaningful and interesting. It was quite informative and thought-provoking. I'm sure that the knowledge shared by you will definitely help us in our future. Once again, I would like to thank you, sir. Thank you so much.
thank you, sir. Thank you very much.
Thank you, sir. So now,
Yes,
moving forward.
you can.
Yes. So now, moving to our third session, we have Dr. Ajit Kumar Thakur. Good morning,
Yes,
sir.
good morning, sir. Welcome to
Good morning, sir.
International.
Good morning.
So he is Dr. Ajit Kumar Thakur from DIPSAR, the renowned pharma school in India. And very at his young age, he's dynamic with his research activities, well popular. And I would like to introduce Dr. Ajit to the Dean of San Pedro, Ma'am Tezro, and the director, Erwin Fowler, sir. They are also in this meeting, and a lot of students from Philippines, San Pedro is also there here watching this international e-conference, and our audiences, the participants, they are from teachers and student and industry, watching from the country and across. So welcome, a warm welcome from Dr. Ajit Thakur. Please, sir, we are already experienced your sessions with our various webinars and orientation programs of the school. I always get all your support in my past experience, and this is
It
also.
is actually our opportunity to get in touch with you all during all the session, and today we will also hopefully will deliver my best in the session.
Please. Yes, Khushbu ma'am, anything you want to say in regards to Dr. Ajit?
Let me introduce,
Khushbu ma'am, hello.
Hello,
Yeah,
sir.
please.
Let me introduce sir to our participants and all the members. Sir is assistant professor of pharmacology in Delhi Pharmaceutical Science and Research University. He has done his Bachelor of Pharmacy from MIT Muzaffarpur, Bihar, and Master of Pharmacy in Pharmacology from IIT BHU Varanasi. He has done his PhD in Pharmacology from IIT BHU Varanasi. Sir has published total 24 research papers, 1 book chapter, 24 review papers, and he has 25 poster and oral presentation in his credits. He has also various awards such as Program Operating Officer of NAMS, GHRE Prize, etc. He is an active member of Indian Pharmacology Society, Indian Academy of Neuroscience, Association of Pharmaceutical Teachers of India, and Indian Pharmacy Graduates Association. We welcome you, sir, on the second day of plenary session of International E-Conference.
Thank you.
Now,
Thank you, Ms. Khushbu. First, a good warm welcome to all of you, and thank you, Dr. Shahu sir, for that opportunity to deliver my research topic in the session of today's international conference virtually in the platform. So I'm going to share my presentation first.
Yes, sir.
I hope it is visible there.
Yes, sir, yes, sir, it is visible.
Yes, it is visible.
Good morning, everyone. So the topic of today's lecture is beneficial effect of glycyrrhizine-rich glycolyzed glabra lean extract on learning and memory in stress-triggered rat. So this is one of the topic we have already completed that project work in our lab at DIPSRU. The glycyrrhizine is one of the major constituent in glycolyzed glabra. We will see why we have selected this plant and why that type of research work we have planned for this. So I would like to say something about the pharmacological efficacy study, some development of the drug, herbal drug development for the neurological disorder. So one of the neurological disorder ongoing in that stress induce some pathology, that comorbidities in brain. Sir has in morning clearly said that there is some herbal product development, there is some target, some parameter you have to evaluate that efficacy, and then obviously the product development. In the just previous lecture, quality by design and quality control in any industry is all related to every parameter, including pharmacological activity of any compound. So that compound came from maybe a source, maybe a plant source, just like glycyrrhizine. So first, I'm going to just say that what is the condition where you have to find a target to have a nudge. So in that work, we have find a stress condition, which is no doubt a homeostasis balance in our body, but in a worst condition, they may affect our several system. So one of the system is central nervous system. So stress is functionally devastating aspect of many neuropsychiatric disorder and neurodegenerative disorder. So the physiological effect of chronic stress means little bit small, which cannot be avoidable, which may be a worst condition develop inside you that negatively affect learning and memory. So what is the link between the stress and memory? We have to explore some target already explored by the scientist, and we have a well-known biomarker, that target, where you can hit your lead compound or a new molecule. The cognitive dysfunction is a major health problem in this current scenario, where lots of stress, lots of complications is there. So in that condition, memory becomes low and that cognitive dysfunction. So according to WHO, 50 million people worldwide living with dementia, and dementia is now the seventh leading cause of death. So somehow we can correlate with the comorbidities of CNS, some link with the dementia and dementia related complications. So that one of the stressor which is linked with the brain, that is the hypothalamus, that is related to pituitary secretion of hormone, that they all relate our body hormonal balances, hormonal function, and the hormonal function also linked with the several cell mediator biomarkers just like cytokines, and ultimately the biological immune system triggered. So it is directly or indirectly stress lead to some complications in our body through CNS, and it also related with the hypothalamic pituitary adrenal cortical axis. That all biomarkers need to be explored well. In our study, we have a rationale of that study that Glycyrrhizia glabra is reported to have antidepressant, anti-stress, antioxidant, and anti-inflammatory activity. So therefore, it might be beneficial for improving learning and memory in stress trigger rat, because we know a stress condition develops these type of changes in the body function, that like oxidative stress going to increase, inflammation going to increase. So may have this effect. So we have a rationale. Then we have a hypothesis. What happened in case of stress, chronic stress? They affect the hippocampus, may cause some shrink in neuronal cell, neurotransmitter modulation in that due to stress related brain function and oxidative stress. So what happened? That lead to neuronal cell death, and that decreases learning and memory. So our target may be a neuronal cell death pathway, maybe some inflammatory pathway. So Glycyrrhizia glabra may have anti-inflammatory activity, which is a known activity that may affect on the neuronal cell death process by inhibition there, antidepressant activity, antioxidant activity. So we have to know the pathway biomarker of that disease, and we can correlate the hypothesis of our proposed study. So the aim and objective of our study was the chronic food shock stress memory deficit model by giving chronic food shock in rat. So for the efficacy study in vivo, we have to take a model. So we have taken the chronic food shock stress model, that is in rat. And the aim of the to study the effect of both extract. We have two extract actually, that from the Glycyrrhizia glabra. One is a normal extract that may be a flavonoid more, and one is extract is glycyrrhizine, the major constituent of Glycyrrhizia glabra. They have developed by the Natural Remedy Private Limited that extract glycyrrhizine rich extract, and we have tested it on the stress rat for that learning and memory effect. The aim of our study also to study the pathophysiology and pharmacology of stress-induced memory deficiency in rat. So we have little bit explored the what is the pathology regarding the stress and that deficiency of memory in rat, and to evaluate the modulatory effect of biomarker, which is known that like cytokines that may be related to antioxidant, maybe some neurotransmitter, which is associated with the stress-induced memory deficiency and target of for further study. So some literature you have to collect for your study. You have to have correlation with the literature available in the global domain, just like many articles are published, many scientific domain is there, you can explore. And then you have some literature review. We have also that we have gone through the plant morphology, distribution, graphical distribution, their habitat, because a plant have to grow in such manner that they have more constituent inside that plant. That is, constituent means active constituent, that is active metabolite, which is having a effect. Just like you have a simple chili, you have a chili, then they have some effect on that. Some region have less bitter taste. Some this type of morphological distribution, geographical distribution effect on the constituent. So this is mainly available in the Asia country, and many species are now grown throughout the country, just like Europe, Asia, and USA. Different activity shows a plant. Similarly, Glycyrrhizia glabra having root, mainly used for that activity. Just I told you, you have literature, so you have to gather all the information where it is having effect. One paper, Dhingra et al. 2004 reported their memory enhancing activity, but they have as such what is the nootropic activity. But this type of activity, we have gone through that stress-induced comorbidity in the first time. So that drug is provided by the Natural Remedy Private Limited, that is the glycorrhea glabra extract. There are two types of extract. One is the flavonoid rich and one is the glycorrhizin rich extract. So that glycorrhizin rich extract having 21.6% weight by weight volume, it is characterized by the HPLC, and the flavonoid rich having less amount of glycorrhizin and more other flavonoids are there. So this is the difference between that. So by this type of method of taking drug, you can compare what is the better efficacious in that nature. For that in vivo study, we have already taken the animal experimental approval by our university institutional ethical committee. That is in for the different type of group we have to take, different group we have taken. One is the control, that is normal, there is no stress, but one is the stress control, means they have going for the food shock stress for that period, and then different drug treatment to that group which having stress. So S plus means they have a stress condition as well as treatment. So different two dose level we have taken for the different extract. One is the flavonoid enriched 50 and 100, and other group is the glycogen rich extract, that is the 50 mg per kg and 100 mg per kg. For the purpose of comparing, we have taken one of the standard drug, that is paracetamol, and we have evaluated the behavioral activity which is related to some memory parameter. We can evaluate behavior basis what is the memory related function, and then we have gone with the some stress biomarkers just like SOD, catalytic lipid peroxidase, and one of the neurotransmitter acetylcholinesterase. That work has been divided for the different group. So that experimental design we have with the different 21 days chronic food shock stress, that is very small current, but it is randomized. That is the 1 hour randomization for the 0.6 milliampere food shock for 0.2 seconds only. So we have randomized in such a manner after 3 seconds, after 4 seconds, and we have minimum 30 shocks per group we have given for 1 hour. And then that 21 days going on, we have also drug treatment for that. And on after 21 days, we have behavior test, that is the treatment plus 1 week behavior study. And then we have different Morris Water, elevated plus maze, rectangular maze, and locomotor activity. These are the well-defined model for evaluation of that memory related activity. And then after that, we have euthanized the animal, collected their liver, collected their kidney, collected their brain for different type of biochemical test and histopathological examination. We have also gone with the statistical analysis, very, very important in the in vivo study, because we have to have correlation with the finding, observation. The chronic food shock model given the scramble, uncontrollable, inescapable 0.6 milliampere shock in electric box 60 times during 60 minutes daily for 21 days. That is the protocol. We have some deviation from that also. Sometimes we have given 50 to 60. In behavioral test, we have find that rectangular maze test, which is the rectangular maze. We have to check the learning assessment by that. That is already defined by the Saxena et al. In that rectangular box, one animal is placed one place. They have to explore another area where there is some feeding material up there. So the rectangular maze is just like a rectangular box. They have pathway, they have to pass from one cabin to other cabin, and they have to reach the last cabin where the food material is there. So this type of learning score, we have to evaluate how much time they have taken, what we have given the score for that animal. We found that a normal rat is not a stress control rat taking more time, means their learning score is more. What it means? They are more time taking for the learning. And a small drug treatment group animal shows less time to get that learning. That is the learning score. So that type of protocol we have designed. So more time taking, that is the learning score is more. But when animal is less time to reach their area, that means it is a learn to go there from one cabin to other cabin and reach to the food material. That is the learning assessment. So stress causes more time to take, and that is called as mode. Similarly, in Morris Watermill test, it is a very well known model that is published in the 1984 only, and many modification going on in that model for the observation of the parameter, the finding. So we have observed day 2 and day 3, the mean escape latencies in that area where they have to escape the water area and go to the platform. And then number of quadrant target, number of entry in the target quadrant where the platform was there, and the time spent in that quadrant to find where it is platform is there or not. So you can think about the situation. If tank is there, there is just 4 quadrant and 1 quadrant having the platform where that animal have to reach there to get escape. Means they have to stop the if they want to be in the platform, so that what is the mean time to get there. So more time taken by the stress control, but after day 2, you can see drug treatment of the glycoregine, mainly the glycoregine rich extract having less time to reach their platform, and they have more time in more entry in that quadrant, because they have learned that this platform was in that quadrant only. So they are exploring their more number of entry in that quadrant, and more time they have had spent on that quadrant to find the platform. Means they have learned more now that this platform is there. So that is evaluated parameter, and we found that glycoregine rich extract having more time spent in that quadrant and more entry in that quadrant compared with the stress controlled rat, which is less. So this type of parameter we can correlate with the treatment group. One of the model is elevated plus maze. That is very known for the anxiety, but you can just modify. That is called as modified elevated plus maze, where you can have a latency to go there. So one animal have to place in the open area, and they have to take time to get inside the area where it is food material is there, or maybe a dark area. So time taken to the entry in the dark area from a terminal portion of the open area is called as latency. Sorry. It means what? They are learning to how go fast to that area where food material is there in the dark field, or simply go to the field where it is the dark, because animals want to be in the dark area only. In open area, anxiety is also there, and they have to learn how to go there. So time taken to go there in the dark area, or to take the food material available in the dark field, open field and dark field, they have to be calculated. That is called as transfer latency. We found that a stress control rat having more time to go there in the dark area where food material is there. Means what? They are not learning much how to so go there with the food material from the open area to dark area. But drug treatment with the glycoregine flavonoid and glycoregine rich, although all are significantly lower in that, but yes, glycoregine having more efficacy compared to glycoregine flavonoid rich extract. That is also a well-defined model for the evaluation of memory related parameter. Locomotor activity we have calculated because some CNS active drug having some effect on the excitation, or you can say some locomotor activity also, that is related to side effect maybe of a muscle function or maybe a locomotion function. So we have gone through that, although this is common in the treatment, but recover from the stress situation, we have found that. So a stress control animal having less locomotor index means locomotor count per minute, and more toward the normal, it is toward the normal rat in the case of treatment group. After that, we have gone with the sacrification and collection of the brain. The brain sample homogenate was prepared in our lab, and the supernatant was taken for the estimation of that acetylcholinesterase and protein, because we have to have enzyme activity compared to protein in that sample. So homogenate of the brain tissue although we have taken the whole brain, but yes, you can go with the hippocampus, you can go with the area that is related to memory. Okay, so in that condition, we have taken the part of hippocampal mixed tissue, not specific to hippocampus only. So we have found that protein level and acetylcholinesterase activity in that manner. So acetylcholinesterase activity, micromolar activity per minute per mg of protein, defined parameter according to Illamens. Okay, so that we have shown that in normal rat and a stress control rat, the enzyme acetylcholinesterase activity is high. What it means? That acetylcholinesterase is going to break the acetylcholine to acetyl and choline. That acetylcholine is a neurotransmitter for memory related function, maybe a nootropic, maybe a cognitive function. We have seen that acetylcholinesterase activity become high in case of stress control stress rat, but treatment group shows less acetylcholinesterase activity, means inhibitory activity of acetylcholine enzyme. So more acetylcholine may be there, and that why the memory is going to increase after treatment with acetyl glycyrrhizia glabra extract. In that condition, you can also see that comparable with the flavonoid and glycyrrhizin rich extract. So more efficacious toward the glycyrrhizin, although it is less than that flavonoid and better than that piracetam as a standard drug. In oxidative antioxidant activity, because we have gone with the oxidative stress, that model, so we have evaluated lipid peroxidation. In that condition, stress control rats shows more lipid peroxidation, and treatment decreases significantly in comparable to both type of extract and standard drug. That another enzyme is superoxidase dismutase. That is one of the enzyme very known. If SOD is more, means your enzyme oxidative property is good. So in case of a stress control rat, SODs become less. So oxidative defense mechanism become weak in that condition, means a stress condition causes less SOD activity in the term of international unit per ml of protein. But treatment of glycyrrhizin extract and flavonoids both have a efficacy in that recovery of that SOD activity. In catalase also the same, just like SOD, we have seen that treatment with the flavonoid and glycyrrhizin extract causes significant increase in the SOD level. Somehow stress causes effect on our organ also. The liver one is the very important. The liver secretes some enzyme which is effective on the other biochemical parameter. Although we have not evaluated that biochemical parameter, but yes, we can. So anatomically wise, it also affects the liver function. In histopathologically, we have seen there is some vacuolar size and their decay in the cellular level component, that is the cell, with the stress control rat. But it is normalized in the stress. That is a representative group as histopathology only, but we can say yes, they have some effect on the liver histopathological area. We have good anatomical histopathological evaluation in the kidney, that normal control rat having good glomerular area, that is the kidney filtration or some kidney related renin pathway may be correlated with the histopathological examination, but we have not evaluated the renin enzyme or renin function. But yes, stress lead to RAS system also, which is ultimately related to cytokines production. So stress control rat having more disturbed anatomical pattern in the kidney, but yes, treatment having normalized toward that. So the summary, the present study provides the evidence for treatment with glycyrrhizin, flavonoid and glycyrrhizin rich extract result in inhibition of behavioral changes induced by the chronic stress exposure. What we have seen that yes, stress causes some behavioral disorientation toward the learning process, but yes, treatment with the extract somewhat it is modulated that pathway, and the normal tendency of learning, you can say memory is increases in case of treatment group. The present study demonstrated that the extract of glycyrrhizin glabra-lin significantly improved learning and memory function and ameliorated the chronic food shock induced oxidative stress impact. So what we have seen that overall study, they have some improved learning memory function, and they also effect on the oxidative stress pathway, because oxidative stress lead to some other enzyme activity also. We have published that paper in the current traditional medicine last year 2020 only, and this is now available in the that journal. Amyloidotic potential of Glycyrrhizia glabra extract on memory impairment in stress triggered rat. So one of the authors is Parul Kambod, who have done that MFARM project under me for that topic, and that paper is published now. So what may be future aspect? We are also working on that, because somehow we have find something, and we have to correlate with the other ongoing work. Nowadays, that is ongoing work is there. We have also targeted some cytokines level in the stress-induced model only, but that time we have taken the model as a restraint-induced stress. Sometimes you can restrain animal, they have some stress effect apart from that chronic food shock. So now we are evaluating that drug in the other model, that is the restraint stress-induced model of that memory function, and we also molecular pathway. So further study are warranted for better understanding of mechanism of action through the molecular and biomarker based exploration of their neuroprotective efficacy of Glycyrrhizia glabra and stress rodent. Some references we have pasted here, but yes, it helped me to get more pointed study planning and that designing the experiment. So I'm thankful to Natural Limited Private Limited Bangalore for that providing that track, and a scholarship from DIPSRU, our university, to MFARM scholar Ms. Parul. She is currently research scholar at Thestis, Faridabad. So thank you. My topic is now over, but yes, you can ask me some question. I will be happy to answer this. I will try to answer your question. Please.
Anyone have any question? Please ask.
Sir, your subject is, I think, so tough. That's why either people may have no idea to ask you any question, or they are quite well educated with this one. Please don't mind him.
No, sir. I will try to have that level of work in our lab so that it can be published in the highly reputed journal. So nowadays, that may be a good news later on, but it is not confirmed yet that one of the project in SCRP with that plant only is ongoing of in the terminal area, actual terminal point only. It may be granted. So we will have more molecular study on that model only.
Thank you, sir. Sir, one thing I have already discussed once with you. So this is a very new university with a new school of pharmacy. I need always some suggestions for the beginners. Yes, anybody, any student want to say something? Some questions? Hello? I think I get some. Dr. Sangram Panda is there. Hello, Dr. Sangram Panda. Okay. Khushbu ma'am, please.
Yes, sir.
Proceed.
Thank you, sir.
दो तो सर एक तो केमिस्ट्री लेते हैं और एक तो फिजिक्स मैथ्स.
Anyone want to say something? Okay, please.
Thank you, sir. Thank you for this wonderful session. We are very privileged to have you, especially me, because you are my teacher. I have learned a lot from you. Thank you, sir. Thank you so much. Now, I would request to our Dean, sir, Dr. Jyotirmay Sahu, to kindly felicitate Ajit, sir, with small token of appreciation.
So, Dr. Ajit, as usual, you always cooperate me, and my sincere regards to Dr. Prabhat Sahu, my teacher.
Yes, sir.
And please accept this small token of appreciation
Thank you.
on
Thank
behalf
you.
of Philippines and Arkazain. Please accept, sir. The memento will be soon sent to you on post.
Thank you, sir. Thank you, Khushbu. Thank you, everyone. Have a nice day.
Thank you, sir.
And welcome, Joseph, sir. Professor Joseph from Philippines. Please welcome. Professor Joseph, we
Good
are unable
afternoon,
to
sir.
see you. We want to see your full face. Can you remove your...
Can you give
please
me
unmute
5 minutes? 5 minutes to
Yes.
program my computer. My apologies.
No matter. We are waiting. In the meanwhile, we will discuss few other things.
Dr. Sahu. Dr. Sahu.
Yes.
This is
Yes,
Dean
ma'am.
Besor of San Pedro College. We are having some connection problems here on our end, but
no,
I think...
Zip my back when I put my... No, not right now.
I am waiting for 5 minutes. No matter. I can wait. A lot of students from Philippines and research scholars and participants are there. I would like to take their name in between. My students are also there. It is too difficult to utter the spellings from my end. That is Padai Hag Vaine. I'm trying. Siega Sekia Kai, Lu Min Cheng, Antija Brian, Babun Maria Angelica, De Asish Ronel, Hannah Patricia Farai, and thank you, dear students and research scholars and participants. You people are encouraging us to do something good for the society, good for the fraternity. And I am seeing you are patiently sitting before the conference for a long hour. It is not so easy task, even for the teachers. Cardona, Maishak, bit difficult to utter because we are not acquainted with this kind of names, ma'am. I'm sorry for if I am doing something wrong. Morales Gabriel, Anna Patricia Farai. So I just want to hear from some of the students who are participating from San Pedro, how their experience. Please unmute your video, and in this small span, you can share your experience to your friends in the India, those who are studying there in Arkazain. Please, voluntarily, one of you, please share your experience. Hello. Please dare to share. Any of my students from Arkazain want to say something in this small time?
Students?
Students, please.
Share your experience. Shobit?
Shobit.
Yes, sir.
Shobit, are you there?
Yes, sir.
Please
Yes,
share
I'm there.
something. What your experience?
Sir, overall, it has been a very informative session. It has been a very interactive session, I must say that. So, we have learned a lot of things, and we have experienced that we have to learn a lot of things, even in a small. Sorry, sir.
1 minute, Shobit. Yes,
Yes, sir.
anybody want to say something? Uh, just a minute, Sobit. One of my
Yes, sir.
friend Ajay Kumar Singh is there.
Yes,
I
sir.
just
Yes, sir.
want to request him to unmute his video. Ajay Singh ji, can you unmute your video? I want to see you.
I will join after
See you.
some time, sir. Please
Okay,
give me
okay.
some.
Yeah. Yes, Sobit, please continue. Sobit, please continue.
Shobit?
Shobit, please continue. Okay. That
Yes, sir. Sorry,
yes,
sir.
please.
So, sir, even from the side of San Pedro College, it has been a very, very good initiative for both of us. We have interacted well, and I hope so. We continue doing such more interactive sessions and e-conferences. That's it, sir. I would like to ask
So
some
if
of
you
the
get
students from
opportunity
San
to,
Pedro to interact with
if
us.
you will get opportunity, hello, Soviet, if you will get opportunity to go for Philippines to do your higher courses, would you
Definitely,
like to
sir.
do that one?
Yes, sir. Definitely, sir. Why not?
Okay. Welcome, welcome, Professor. Welcome, Professor Joseph.
Am I
Now,
audible?
I think,
Yes,
yes,
sir, you're
yes, you
audible.
are nicely audible. You are nicely audible.
Thank you. My
So welcome to
apologies
India.
for the slight delay.
No problem, sir. So welcome to India.
Thank you.
And I definitely like to see you people, ma'am, Tezro and Erwin Fowler, sir, and you, particularly Joseph. Please,
It's our pleasure,
once
sir.
visit our, once visit our university. And as I have committed with you yesterday, I just want to see one movie with you people, Indian movie on the basis of friendship.
Oh,
So,
that
Kushbu
would be
ma'am,
great.
please carry out.
Yes, sir. So now for our fourth session, we have Professor Joseph, Assistant Professor in Pharmacy Department, San Pedro College, the Philippines. Sir has done his BS Pharmacy from San Pedro College, Philippines, and MS Pharmacy from University of Santo Thomas, Philippines. He is a full-time faculty member and academic research coordinator, Pharmacy Department in San Pedro College, and a regular member of Research Ethics Committee of San Pedro College. He is associate member of American Society of Pharmacognosy and also appointed as continuing professional development monitor council of pharmacy professional regulation commission from 2019 to 2021. A very good afternoon, sir. I welcome you on behalf of School of Pharmacy, Arkajan University for this plenary session. Now may I request you to kindly lighten the session with your valuable words. Over to you, sir.
Thank you for that warm introduction, ma'am. To the honors of Arcajain University as well as San Pedro College, to the students who are inside in this conference, participants, ladies and gentlemen, good afternoon. I am delighted to present to you one of the findings of one of my recent scientific productivities that involves certain determination of the safety and efficacy as well as establishing the bioactivity of a native plant that is found here in the Philippines and some countries in Southeast Asia. Okay. In ancient times, people before would really utilize plants as medicinal agents, even in which these medicinal agents that come from plants or could be taken as food sources as well. This is very evident in one of the oldest medicinal philosophies of the father of medicine, Hippocrates. One of his greatest medicinal philosophy would be, let food be thy medicine and medicine be thy food. So from this premise of Hippocrates, we could really ponder or reflect that medicinal agents could really come from plant sources or even plant species that are being utilized as food, or it is taken or ingested as food. Up to this date, our societies is in quest for finding commodities or agents used to treat or manage diseases or ill conditions, which are very common to people in different countries. So these examples of diseases would be hypertension and diabetes mellitus. So one of the commodities that could treat these metabolic related disorders would come from plants that could possess medicinal compounds. Okay? These are also called as active phytochemical compounds. So a number of plants in which these plants are being classified into different plant families, different plant genus, up to its characterization of its species. So an example of a plant genus that would also contain medicinal plants would be the genus of Artocarpus. Okay? As reported in the literature of Jagtap and Bapat, those plants that belong to the Artocarpus genus contains polyphenolic compounds which have medicinal value. An example of a plant species that is under this genus is Artocarpus odoratissimus, or we call it here in the Philippines as marang or marang fruit. Since there are limited investigations about this native plant, this scientific work aimed to assess first the pharmacognostic properties of the plant, specifically the fruit sample of the plant, in which its safety and purity as a medicinal agent was ascertained. Next would be the characterization of plant compounds, specifically polyphenols that are present on the plant. And this work also established the bioactivity of the plant species utilizing in vitro and in vivo techniques. So this slide shows the workflow or the methods that are being carried out in this study. So this study would involve 4 phases. Okay, 4 phases. The first phase includes plant sample collection, species authentication or verification of the plant that is being collected. A botanist will be was the curator who verified the plant sample that was collected, and drying of the plant material using lyophilization process. The second phase now involves one of the rate limiting steps or procedures in this research endeavor. It involves determination of the pharmacognostic properties of the plant sample. Okay? And here are the following pertinent parameters that are utilized to the testings. Then third phase is attributed for the main plant extraction process, in which once the crude extract that was attained from the main plant extraction process will be subjected to fractionation in order to attain different solvent fractions, or it is usually called as semi-crude extracts. And also included in phase 3, the third phase of this research work, is the analysis of the phytochemical compounds that are present in the extract samples, utilizing different techniques. Lastly, or finally, the fourth phase or the final phase of this research project is all about the establishment or assessing the bioactivity of the plant, specifically its blood glucose lowering or its hypoglycemic bioactivity. So on the plant collection, the plant samples were collected specifically here in a city, a key city here in the Philippines, Davao City. The samples were collected in an early morning time. So the principle of, or the reason why do plant samples needed to be collected in the earliest set of time in a day? It's because that the intensity of the sunlight is not that too much high, because in natural products research, you need to protect plant materials containing active compounds from disruption, in which high intensity of sunlight could also disrupt the integrity of the plant compounds present on plant materials. The totality of the plant samples that were collected were processed and stored in a suitable environment. And a part of these plant samples that was collected were submitted as a specimen for plant species verification or authentication, plant sample species authentication, to a botanist curator, in order to prove that that collected plant sample is really the species, the right species. And right after this set of preliminary processes, the collected plant sample was also subjected to drying, specifically lyophilization drying. So there are different ways in order to dry plant materials. We could do air drying or vacuum drying. Okay? One of the methods for vacuum drying would be lyophilization process. But we need to take note the factors needed in order to choose an appropriate drying technique for plant samples. Okay? So since the plant sample that was collected in this research endeavor was a fruit plant sample, thus it is not ideal for air drying because it would be subjected to a lot of possible contaminations. So the appropriate technique in order to dry fruit plant samples in general would be vacuum drying or the lyophilization process. So these are after drying the plant sample, the next phase of the research project was also conducted. So the second phase, like what I've mentioned earlier, it's all about the determination of the pharmacognostic properties of the plant sample. Okay, So as stated in this PowerPoint slide, so these are the following pharmacognostic parameters that is observed and analyzed in the collected plant sample. Okay, what are these? We have the ash content, the extractive value, the moisture content, the microscopic observation of the sample, as well as its fluorescence testing. Okay, so each type or each parameter has its own principle and importance. And that would be my next discussion. Let's first discuss about ash content analysis. Okay, so in conducting ash content analysis, an amount of the plant sample that you dried must be placed inside a crucible. Then you must subject it to high temperature heating or incineration at 500 to 600 degrees preferably. Then right after some time, there would be an ash macroparticles that would be present, if any. This ash macroparticles indicates the presence of impurities like carbonates, oxalates, and silicates, in which this could also be a parameter to describe the purity of your plant samples for crude drug plant processing, or crude plant drug processing rather. Okay. Once if an ash macroparticle is present, so an alikot of that ash will be dissolved in water. And once it dissolutes, it would be called as the water-soluble ash. So the indication of this water-soluble ash is used to estimate the amount of inorganic entities or compounds that is present in the sample. Okay? Another alikot of that ash, okay, that ash macroparticle must be reacted with an acid solvent like hydrochloric acid. If in case the ash material won't totally be dissolved in the in the acid solvent, again, if that ash macroparticle could not be dissolved in that acid solvent, then it would be called or termed as acid insoluble ash, in which this acid insoluble ash represents silica that is present, in which it pertains, rather, the presence of silica would pertain to a possible contamination of the plant sample that is being subjected to crude plant drug processing. So on this research study, the plant sample, or the fruit plant sample of Artocarpus odoratissimus demonstrated a very minimal ash contents. Next parameter for pharmacognostic property testing is the extractive value. Okay? So the extractive value determines the amount of the active compound in a given amount of a plant material once that this small alikot of the plant material is soaked or reacted with a particular solvent. So this analysis gives an idea of the appropriate solvent to be used for the main extraction process. Okay? Again, I would like to reiterate that this parameter would also give a concept, an idea for laboratory analysts. What is the appropriate solvent to be used for the main extraction process? So how does this extractive value determination is done? This is performed usually with a small amount or an aliquot of the plant material and soak it with a specific solvent. Then subject that in agitation using a mechanical.
हाँ यूट्यूब में है बाबू.
Okay, so on the slide, I would like also to just show this is the one of the mechanical shakers that we have in our institution. So on this research endeavor, I utilize this as one of the equipments to do the agitation procedure that would last for 4 to 6 hours. Then after that agitation process, it will be subjected to solvent evaporation, or it will be dried under vacuum at 45 degrees Celsius. Then the matter that would be left will be weighed. And it is also a variable. The weight of that matter left is a variable in computing extractive values. On this study, we utilize 2 solvents. We have distilled water and 95% ethanol. So the dried plant material was subjected to extractive determination, extractive value determination for distilled water and for 95% ethanol. So among these 2 solvents, the plant sample yielded a very significant extractive value on 95% ethanol. Thus, ethanol, an alcoholic solvent, will be utilized in the main extraction process that I will be explaining to you later on. Another important parameter for pharmacognostic analysis is moisture content. Okay? So moisture content of plant samples determines also the purity of plant samples. So it should be at a minimal level because once that it exists on a significant level to plant samples, okay, it pertains to microbial, bacterial, or fungal contamination of the plant sample. That might be a precursor for microbial contamination. So higher amounts of moisture can really attract contaminants to the plant material or to the plant sample. Guidelines of the United States Pharmacopoeia or USPNF has a limit of 10%. That moisture content in any powders or any substances should not be more than, solid substances rather, should not be more than 10%. So in this research study, we utilized a digital moisture analyzer, which is also available in our laboratory at our institution. So usually the inside of this digital moisture analyzer, an aliquot or an amount of that dried plant sample is placed inside. And you will just close it and automatically this equipment would read its moisture content. Okay? The result on this study showed that our plant sample, the plant sample utilized in this study, has a very minimal moisture content, which is less than the limit from USP, which is 10%. Next is the microscopic analysis or the microscopic characteristics of the plant sample. The microscopic analysis is utilized to observe the integrity of the tissue structure of plant sample. So it utilizes different staining reagents, and the sections were affixed in slides, and then staining reagents will be incorporated. And upon completion or processing of these slides, it can be viewed with the aid of a microscope. In this study, the plant sample undergone staining using methylene blue and counterstained with safranin. Okay? And one of the final processes in the slide preparation is mounting. So the substance that was used for mounting is Canada balsam. Then after the microscopic slide processing, it is subjected to observations under a microscope. In high power objective, that is also represented in this slide. In under high power objective mode, the plant tissues of the plant sample upon observation contains thin-walled cells densely packed with granules. And some of these granules occurs as well-defined clusters. So meaning to say that the plant tissues of this plant sample have a good integrity, or it has a good characteristic, and it cannot be easily disrupted. Okay, the last parameter for the pharmacognostic property testing is fluorescence analysis. Okay, so fluorescence analysis is used to preliminary characterize presence of phytochemical compounds, because these compounds, by principle, it absorbs light, because these are active compounds and they absorb light. And they would also display different ranges of colors upon exposure to a specific wavelength of light, as reported in the literature of Pimpel et al. In 2012. Some of the phytochemicals or phytochemical compounds exhibit fluorescence in certain reagents or solvents. Okay? So once that you add a specific reagent or substance to a plant material or plant sample, definitely it would exhibit color formation. There would be a color formation after you reacted that substance with the plant material. Okay? Fluorescence is the phenomenon exhibited by these plant compounds present in the plant samples or plant material. And this fluorescence color is very specific, or it pertains to a specific plant compound. In this study, the dried plant sample was placed on a microscope slide, and 1 to 2 drops of a specific reagent or substance, or a chemical reagent rather. Okay? That powdered plant material is reacted with a specific reagent, and it manifested a specific color under observation on a visible light, a short ultraviolet light at 254 nanometers, and a long wavelength addressed at 365 nanometers. So there are a lot of chemical reagents that we utilize. So this color that is being produced that would fluoresce in the presence of light manifests presence of phytochemical compounds. So after the pharmacognostic properties evaluation of the plant sample, crude plant extraction process was conducted using maceration. So by principle, we know, or we learned, that there are two general crude extraction process, maceration, and percolation. So these two crude extraction process has the same principle. The principle of soaking. Soaking the plant material with a extracting solvent. So in this study, in this research endeavor, we utilized maceration technique using 95% ethanol. Okay? I forgot to discuss the difference between maceration and percolation is the medium that is utilized in the extraction process. And there is one of the advantages of using percolation is also the flow of the solvent upon filtration process or attaining the liquid extract. But for compounds that are being attained after the extraction process, there is less significant differences between the two. Okay? So in this research endeavor, we utilized maceration process. And from the result of the extractive value determination, since the alcoholic solvent or 95% ethanol has a good extractive value with the plant material. That's why it is being chosen as the extracting solvent in the maceration process. Okay? And the soaking of the plant material was conducted for 24 hours only. Okay? It did not exceed the 24 hours period. Why? Okay? Literature of Sarker et al. On natural products research would tell us that soaking the plant material for 24 hours or not more than 24 hours could preserve the structural integrity of these active compounds that are present on the plant sample. Okay? So only 24 hours, soaking for 24 hours. Then after the maceration process, the liquid extract was dried using a rotary evaporator equipment, or it is dried under vacuum at 45 degrees Celsius. Then a viscose extract was attained. So the percentage yield that was attained on the extract that was collected was 18.54%. So what are the variables needed in order to compute for the percentage yield of your crude extract? You need the weight of the dried powdered plant material used for the extraction process, as well as the weight of the viscose extract that is being collected. So that these are the two variables that are needed to compute for the percentage yield. Okay. Then right after the crude extraction process, a significant amount of the crude plant extract was subjected to solvent fractionation following Kupchan's solvent partitioning scheme with modifications. Okay? Kupchan's partitioning scheme is one of the standard fractionation methods that are utilized, preliminary fractionation methods utilized. Okay? The Kupchan solvent partitioning scheme. So the process of this followed the scheme of Kupchan with modifications in which the aliquot, the significant aliquot of the extract was dissolved in water. Again, the significant aliquot of the viscose extract was dissolved in water in order to make or to form an aqueous suspension of the extract. This aqueous suspension of the extract is placed in a separatory funnel, okay, which is represented in the slide. Its graphical representation is on the slide. The separatory funnel containing the aqueous suspension of the extract. And eventually, it is partitioned or fractioned using different solvents of increasing polarity. The solvents that was chosen was hexane to represent the non-polar, so the group of non-polar solvents, dichloromethane as the semi-polar solvent, and butanol for the polar group of solvents, and butanol for polar group of solvents. Okay, so it was partitioned. So usually in the partitioning scheme, the first is the aqueous suspension is now in the separatory funnel, and an amount of the non-polar solvent, hexane, for example, is placed in the separatory funnel. Then it would be agitated slowly, and the lower layer, which represents the lower layer would represent the aqueous, and the upper layer is the hexane layer. And the upper layer is being collected, and the lower layer, the upper and the lower layer are being collected separately through the use of separatory funnel. And the aqueous layer will be placed again inside the separatory funnel, and it would be subjected to another partitioning with dichloromethane, then after dichloromethane with the polar solvent butanol. Okay, so that is the partitioning process. So these liquid extracts or solvent fractions or liquid extracts were subjected to drying at under vacuum again for 45 degrees Celsius. And viscous extracts are now collected. So these viscous extracts, the hexane extract, the dichloromethane extract, and butanol extract are termed as semi-crude extracts. Okay? So one of the purpose of this solvent fractionation or solvent-solvent partitioning is to preliminary separate plant compounds based on their polarities. Just simply basing on the principle that like dissolves like, because different groups of plant compounds has certain polarities. Some of them are very soluble with polar solvents. Some of them are soluble with non-polar solvents. So this solvent fractionation is a good avenue for preliminary or initially separating plant compounds based on their polarities. Okay? After attaining these semi-crude extracts, these extract samples together with the crude extract were subjected to phytochemical screening analysis. Okay? The target for this phytochemical screening analysis would be the polyphenols, because most of the plant species under Artocarpus genus are very rich in polyphenolic compounds. For characterizing the presence of polyphenols, this study utilized first thin layer chromatography. Actually, this study utilized two chromatographic techniques in order to qualify polyphenols. First would be the thin layer chromatography and the high performance liquid chromatography. So the principle of chromatography is based on the separation of compounds, characterizing a specific compound based on separation principle. Okay? Again, the principle of chromatography is based on the separation of compounds in order to determine the presence of a specific group of compound or a compound or a constituent present on a sample mixture. So on the thin layer chromatography process, a silica gel plate is utilized. So that is the stationary phase in which the separation of compounds which would be evident in the formation of spots is being present on the plate. Okay? The manifestation of the separation is evident on the silica gel plate in the thin layer chromatography. You just need a solvent system, or solvent system is usually termed as mobile phase, that causes the separation of these different compounds. And these different compounds are represented as a spot in the silica gel plate. So after the process run on the chromatographic plates, these were observed under ultraviolet light, and the silica gel plates were sprayed using different visualizing agents. So these are the examples of these visualizing agents that would really indicate presence of polyphenols and other polyphenol compounds like flavonoids. Okay, so there is a specific color of the spots after spraying this visualizing agent to the plate. For potassium ferricyanide ferrichloride, usually blue spots would appear after spraying or after exposing this silica gel plate containing the spots with this visualizing reagent. For antimony chloride, orange spots would be evident on the plate after spraying the plate with antimony chloride. As for vanillin sulfuric acid, violet or light red spots are evident to the plate. Okay, so this table would summarize the number of spots that are present on the different silica gel plates that we utilize in our thin layer chromatographic analysis. Okay, for the high performance liquid chromatography, okay, only the semi-crude extracts were subjected to high performance liquid chromatography. Okay? High performance liquid chromatography is an analytical method used to separate as well as distinguish a compound in a mixture. So it has a better accuracy when it comes to characterizing compounds. Okay? So there are two types of, or two modes, or two types of techniques of high performance liquid chromatography that would be based on the mode of elution of the mobile phase or solvent systems. The first is the isocratic technique, okay, in which the solvent system or the mobile phase mixture is consistent as well as its flow rate is consistent as the analysis would go through. Okay? Common drug products that we have in the market now are analyzed through isocratic technique. On the other hand, the other type would be the gradient analysis. Gradient analysis is very useful, or it is very efficient for analyzing sample mixtures with unknown compounds. Okay? An example of that sample mixtures with unknown or uncertain compounds are plant extract samples. Okay? This gradient analysis, what happens to this is the mixture of the solvent systems or the mobile phase is changed during measurement, and it influences the retention of compounds. And there is a gradual flow of the mobile phase as the analysis goes through. The one that is ideal for plant extract samples would be the gradient mode of analysis. So in this research work, gradient technique was used with two sets of solvents. Okay, so the two sets of solvents. As the mobile phase that I used was acetic acid and a combination of acetic acid, a lower concentration of acetic acid and acetonitrile with a flow rate of 10 microliters per minute. Okay. So after the individual chromatographic run process of each semi-crude extract, the chromatogram, only the chromatogram of the dichloromethane or the DCM semi-crude extract demonstrated 4 prominent peaks in retention time, by which the first prominent peak, which has a retention time of 7.1, is comparable with the positive standard that I utilize. Okay? We utilize quercetin as a positive. We also run a standard using this analytical parameters, analytical mode of high performance liquid chromatography. Okay, so upon comparing, so this first peak is much more comparable with the peak that is present on the chromatogram of the standard, which is quercetin. Quercetin is a flavonoid that is classified under polyphenolic compounds. For quantifying phenols and flavonoids, flavonoids are examples of polyphenolic compounds. In order to quantify phenols and flavonoids, the method that we utilized was the UV-vis spectrophotometric method. Okay? So the main principle of this method is based on the active compounds that readily absorbs light. And by light absorption, the amount of light absorbed is proportional to the amount of compounds present on a sample mixture. For total phenol content determination, Follin-Shukaltu method was used. So this method, okay, or basically the reagent on this method contains metallic compounds, because these metallic compounds or this reagent containing metallic compounds would bind with the phenols present on the sample mixture. It would form metal complexes. Okay? By complexation reaction, it would form metal complexes in which this could be observed or measured using absorbance in spectrophotometric technique. For flavonoid content determination, the sample mixtures or the extract mixtures were reacted first with aluminum chloride. By principle, aluminum, the element aluminum, readily binds or it is highly reactive with the nucleus of flavonoids. Okay, the nucleus of flavonoids is benzopyrene. So it definitely, or it readily binds to the fourth and fifth carbon atoms present on the benzopyrene nucleus, the nucleus of the flavonoids. And this binding or reaction is manifested in the formation of metal complexes, in which it can be quantified using absorbance in a spectrophotometric analysis. Okay, so for total phenol content determination, the standard that was utilized was gallic acid, and the standard for the calibration for the flavonoid content on total flavonoid content determination was quercetin. Okay, so results showed, as presented in the PowerPoint slide, results showed that among of the sample extracts, the crude and the dichloromethane semi-crude extract demonstrated a considerable phenol and flavonoid content expressed in gallic acid equivalent. For total phenol content assay, and quercetin equivalent per ml of sample for total flavonoid content assay. Again, the DCM semi-crude extracts among all of the semi-crude extracts exhibited considerable or a very good amount of concentration of both phenol and flavonoid utilizing spectrophotometric method. Okay, so that ends the third phase of this research endeavor. Now moving on to the fourth phase or the final phase of this research project, it involves bioactivity testings. Okay? It involves in vitro and in vivo bioactivity assays. Okay? So in the in vitro test, we utilize the alpha glucosidase inhibitory enzyme inhibitory model. Again, in the in vitro test, we utilized alpha glucosidase inhibitory model, alpha glucosidase enzyme inhibitory model, because by principle, the role of alpha glucosidase enzymes, it catalyzes, this enzyme catalyzes the breaking down or the disruption of oligosaccharides that comes from polysaccharides, which are present in food that we eat, or the starches that we eat, okay? Or the starch containing foods that we eat, okay? In which it could be broken down into oligosaccharides, and these oligosaccharides can be broken down into monosaccharide units with the aid of the enzyme alpha glucosidase. So this alpha glucosidase enzyme now is one of the targets, novel targets, in order to decrease blood glucose levels. What might happen once that these oligosaccharides would be broken down into monosaccharide units like glucose? So this glucose can be readily absorbed in the bloodstream. And in effect, it will cause increased blood glucose levels in a physiological system. Thus, inhibitors of these enzymes of this enzyme alpha glucosidase is one of the novel targets in order to decrease blood glucose levels. Okay? So, in this in vitro assay, like what I've mentioned, alpha glucosidase is one of the main substances or reactants that is involved. Okay, the substrate for it is PNPG, or the para-nitrophenyl glucopyranoside. Okay? So these two are the important reactants in the assay, in this in vitro assay. And the positive control or standard that was utilized is acarbose. Acarbose is an established alpha glucosidase inhibitory drug. So all of the extract samples in this research endeavors, all of the extract samples were subjected to alpha glucosidase inhibitory assay. Okay, so amid all of the semi-crude extract samples, the semi-polar or the dichloromethane semi-crude extract exhibited enzyme inhibitory activity, in which the parameters that are used to compute for the inhibitory activity would be in percent inhibitions. Aside from percent inhibitions, this percent inhibitions, we could also deduce an IC50 value. So what is this IC50 value? So IC50 values are computed, okay, that are based from percent inhibitions of the respective concentrations of the sample. So the inference of IC50 value is the lower the IC50 value, the lower concentration of the sample or mixture containing a compound need to inhibit 50% of the total quantity of the enzymes present. Again, the lower concentration of a sample, okay, the lower the IC50 value, rather, the lower the IC50 value, the lower the concentration of the sample or compound needed in order to inhibit 50% of the total quantity of the enzymes present in the reaction environment. So this pertains also to the potency of the sample. Among all of the samples, including the positive control, okay, yes, of course, a carbos would really have a good IC50 value. Okay? The one that is comparable to its IC50 value is the dichloromethane semi-crude extract. Okay? So what could might be the interpretation of this? Okay? Since phytochemical screening is already performed, it was proven that polyphenols are present in dichloromethane semi-crude extract, both from chromatographic and spectrophotometric methods. So an interpretation that I can deduce is the one that is responsible for its considerable alpha-glucosidase inhibitory activity would be the presence of the polyphenolic compounds in the dichloromethane semi-crude extract. Okay. Since the dichloromethane semi-crude extract exhibited a good in vitro alpha-glucosidase inhibitory activity, then this was utilized in the in vivo testings. Okay, prior to the conduct of the in vivo biological test, okay, animal research permit was first attained from the Bureau of Animal Industry or BAI, Philippines. Okay, because in our country we cannot directly perform biological testings involving animal models if we don't have this permit. I think it also applies with other countries as well. Okay, because there would be ethical implications if you do not seek or attain an animal research permit from the authorities of your respective countries. Okay, again, prior to the, just a disclaimer, prior to the conduct of the in vivo testings, animal research permit from our Bureau of Animal Industry was attained. Okay, so there are two in vivo tests, in vivo biological testings that was conducted. First would be the acute oral toxicity test, because the result of the acute oral toxicity test would be the basis for deducing the doses of the plant extract sample that was used in the hypoglycemic assay using rodent animal models. Okay, so for the acute oral toxicity testing, the Organization for Economic Cooperation and Development Guidelines, or OECD guidelines, was used for oral toxicity tests. So there are two general types of this OECD guideline oral toxicity test. We have the limit test and the main test. Okay, the limit test is usually used for samples. Okay, having toxicity profile. Okay? If your sample has already preliminary toxicity information, then and you just want to verify its toxicity, then limit test can be used. Okay? Limit test is the appropriate type of OECD test for it. But if in case your sample to be tested has very, very limited toxicity information, or there are no toxicity investigations about your about the sample, then the main test is really appropriate, or it is designed for the sample having less or limited investigations on its toxicity information. Since the plant sample, Artocarpus odoratissimos before, has very less information about its toxicity. That's why we performed the main test from OECD guidelines. Okay, so briefly on this OECD 4 to 5 main test, 6 female Sprague dolly rats were used, and includes also a normal control test rodent. So this OECD main test is an up and down dose procedure of toxicity test. So the doses that was utilized based on the guideline was 175, 550, 1,750, and 2,000 milligrams per kilogram body weight of the extract was used. So these were the doses based on the guidelines. And the test animals were subjected to fasting overnight before administering the extract. So the mode of administering the extract was through the use of an oral gavage. Okay? The first set of test animals was administered with the very low dose, 175 milligrams per kilogram. And when the animals survived after 48 hours, to the next animal, test animal, the dose was increased, okay, with a factor of 3.2, which was 550 milligrams per kilogram. If that second animal, when the second animal that was administered with 550 milligrams per kilogram survived, administered with 550 milligrams per kilogram, survived after 48 hours, then the remaining animals will be subjected to a higher dose. Now, what the highest dose was 1,750 and 2,000 milligrams per kilogram dose. Okay? And the testing was ended until the last 3 animals. Those 3 animals that were administered with the very high dose, 2,000 milligram per kilogram, survived after 48 hours and then after 14 days. Okay? Then you conclude that the plant sample or the plant extract sample is fairly non-toxic, up to 2,000 milligrams per kilogram. The highest dose or ceiling dose on the OECD guideline was 2,000 milligrams per kilogram. Okay? So after the observation, if the animals survived or some test animals died, since all of them survived, okay, they were subjected to gross necropsy procedures. And the gross necropsy procedures was performed by a licensed veterinarian. Okay? Results on this study showed that the gross necropsy findings based from the observation of the licensed veterinarian It revealed that all of the essential organs like the liver, kidneys, lungs, and the heart were normal, possessing a firm consistency. Okay? Then we performed subacute toxicity testing, specifically on the liver and the kidney organs of the test animals. Okay, so there were no, we isolate the liver and the kidney organs and subjected it to histopathological examination. So in the histopathological examination, a veterinarian histopathologist interpreted the results, and results showed that there were no signs of proliferation and no signs of congestion of blood vessels that were noted or being observed. Okay? To sum up the results of this acute oral toxicity study, the extract sample is fairly non-toxic up to 2,000 milligrams per kilogram of body weight of test animals. So this was the basis for the dosing. In deducing the low, medium, and high doses of the extract sample that was applied in the in vivo hypoglycemic bioassay. Okay, so let me discuss to you the rodent animal bioassay parameters or conditions that was. So there were 236 male sprog dolly rats. Why male sprog dolly rats? Because male species of rats does not undergo any hormonal fluctuation, since the bioassay is all about a certain metabolic disease. Okay? Hormonal fluctuations that are being that are occurring rather in female rodent species could also interfere results. That's why results in the bioassay. That's why male species of sprog dolly rats were utilized. Okay, 36 male sprog dolly rats were randomly or allocated in 6 experimental groups. Okay, this is represented in the table that is found on the PowerPoint slide. Okay. So we have the normal control group. Okay, so the normal control group didn't receive any extract or any standard drug. Okay. Next is the negative control group. The negative control group, okay, this animals that were allocated in this group were only induced with hyperglycemia, but there were no drug that is given to them. Only the diluent of the extract. Again, only the diluent. Solely the diluent of the extract is administered to the negative control group or to the animals allocated in the negative control group. Okay? For the third groups of animals, they were administered with the positive control drug, which is acarbose, dosed at 10 milligrams per kilogram. The final group allocations, the 4th, 5th, and the 6th group allocations, these are the extract treated groups of test animals. Okay? So the 4th group, in which the animals allocated to it are given with a low dose of the plant extract of the extract sample. The 5th group were administered or given with a medium dose of the plant extract. And lastly, the last group in which the animals are being allocated to it were orally administered with a high dose of the plant extract. Okay, so the doses were based from the 1/8, 1/4 recommended dose levels of the recommended dose levels that came from the maximum dose of the acute oral toxicity testing. Again, the doses were based again from the ceiling dose of the acute oral toxicity testing, 1/8 of it, 1/4 of it, as well as 1/16 of it to add to the maximum dose or the ceiling dose of the acute oral toxicity testing. Okay? And the inducing agent for hyperglycemic condition to the test animals was alloxan trihydrate. Okay? It was administered at 150 milligrams per kilogram through intraperitoneal route. So before the induction of hyperglycemic condition to the test animals underwent 18 hours of fasting. Okay? One of the mechanisms why alloxan trihydrate would really induce hyperglycemia because it targets the islets of Langerhans present on the pancreas, okay, in which this is involved in the production of insulin. So basically, alloxan trihydrate impairs insulin production. Okay, that is why it is one of the choices for inducing hyperglycemic conditions to test animals. Okay, so oral administration of the extract sample as well as the positive control acarbose took for 21 days. Okay, and blood extraction or measurements of blood glucose levels were done through blood collection on the tail vein of the test animals. And prior to blood collection, they were also undergone fasting overnight. They undergone fasting overnight. Okay? And the frequency of measuring blood glucose levels were conducted 5 times. Okay? So I mentioned earlier. We have the baseline measurement from the start after the acclimatization period of the test animals. The post-induction, okay, after they are being induced with hyperglycemia. Okay, this could also note if the animals or the test animals are really in hyperglycemic conditions, which is evident in their blood glucose levels. Then after the post-induction measurement, extract treatment as well as a positive control were given to their respective assigned groups of allocated animals. Okay, so treatment, oral treatment or oral administration of the extract samples and the positive control took for 21 days. And in every 7th-day frequency, the first 7th day, the blood of the test animals were measured. Okay? On day 14, on the 14th day of administration, the blood glucose levels were also measured. And lastly, the 21st day, that was the last duration of attaining or measuring blood glucose levels in the test animals. And blood glucose levels were biochemically measured and analyzed using a liquid color kit through spectrophotometric technique. Okay, so the blood glucose levels of the test rodent animals is utilized as the essential biochemical parameter in the bioassay, like what I've mentioned earlier. And the mean blood glucose levels were measured in all test groups of rodents on the baseline, the induction time, day 7 treatment, 14th day of treatment, and 21st day of treatment. Okay, so the negative control, the negative control group, okay, there were no decrease of blood glucose levels that were noted or measured, because basically there were no substances that are there were no drug treatments, even the extract or the positive control. It was not given to that treatment, that group of test animals, so the negative control. Only the diluent of the extract is given to them, which is twin 80 in normal saline solution, which is an inert mixture that is a good diluent for plant extracts. Okay, so aside from that, decrease of blood glucose levels were noted in day 14 measurement after the day 14 treatment. Okay, so specifically, the groups, the last treatment group, or the group that was given with a high dose of the plant extract has a comparable blood glucose lowering activity with the positive control group, which was given with acarbose dose at 10 milligrams per kilogram. Again, the high dose, the group of test animals that was treated with the high dose of the extract exhibited a blood glucose lowering activity after 21 days of giving or administering the extra sample. It exhibited comparable activity with the test group given, or the test group of animals given with the positive control, which is acarbose. So the results indicates that basically the high dose of the extract can have or can possibly have a comparable activity with the positive control in decreasing blood glucose levels. Okay, the weights of the test animals were also noted. So upon induction of hyperglycemia from the negative control group up to the group of test animals given with a high dose of the plant sample extract. Okay, during the could observe in the graph that during the post-induction period, or after they are induced with hyperglycemia, most of their weights decrease, because one of the manifestations of having diabetes mellitus is a decrease in weight as well. Okay, and there were also improvement of weights among the extra treatment groups were observed as the administration of the extra pushes through to 7, 14, and up to the 21 day. Okay, so in conclusion, Artocarpus odoratissimus fruit sample basically passed the limits of the pharmacognostic analysis or parameters needed to prove its pharmacognostic properties, and as well as the results of this pharmacognostic property testing could serve as a preliminary establishment of this plant species to become a medicinal plant. And based on the phytochemical screening procedures, it possesses polyphenolic compounds which might be responsible for its substantial in vitro and in vivo bioactivity. And based on the toxicity studies, it is fairly non-toxic, up to 2,000 milligrams per kilogram body weight of test animals. For future directions, or what is the main significance of this research project in our countries or in our communities? Okay, so for future directions, or if some researchers would also use this study as one of their concepts in order to formulate new ideas or new researches or new research studies. Okay, so it would be that the other plant parts of this plant species would be considered for analysis, and in-depth screening of the other types of phytochemical compounds present on the plant might also be considered for future directions. And as for the significance, one of the scientific significance of this research work would be, this would be a source of information about the medicinal value of this plant species. Actually, the fruit of this plant species is commonly cultivated or propagated for recreational purposes, or as a gift that is given to your friend or to your relatives, or basically it is a fruit that is used for recreational purposes. Okay? It is one of the significance of this study is to prove that this plant really has a medicinal value. Aside from this fruit, the fruit of this plant is popularly utilized as a recreational fruit, or a fruit that is used for recreational purposes. Okay, and again, this study would serve as a basis of information about the bioactivity or the medicinal value of this plant for those people who cultivate or propagate this native plant species, most especially in Southeast Asia, in which the fruit of this plant is not just a local delicacy, or it is not just only consumed for recreational purposes, but it is a safe and an efficacious medicinal plant material. So before I end my presentation, allow me to acknowledge the National Research Council of the Philippines, the Philippine Council for Health Research and Development of the Department of Science and Technology, which supported this research endeavor or this research project. They financially assisted the completion, up to the completion of this research project. And I would like also to acknowledge my partners in this research study, Ms. Kay Ann Honatas and Dr. Ross Vasquez. And I am Joseph Marie Querequinsha, and I wish all of you good health. And together, let us rise from the fetters of this COVID pandemic. Once again, good afternoon and good day to all.
Thank you, sir. Thank you for this wonderful presentation. It was very informative. Now, I would like to request Dr. Babita Sarangi, Associate Professor, School of Pharmacy, Arkajan University, to kindly felicitate Professor Joseph with small token of appreciation.
Thank you, sir. Thank you, sir. Thank you for joining us and gracing the event with your benign presence on virtual mode. Thank you. At this juncture, am
Indeed,
I
my
audible,
pleasure.
sir?
Yes, yes, you are audible.
At
Yes,
this
Professor
juncture,
Joseph.
I
Yeah,
extend
tell
my
me.
heartfelt thanks to our honorable speaker, Professor Joseph, Pharmacy Department, San Pedro College, Philippines. Thank you for making an excellent presentation, sir, and making this session interesting. The knowledge shared by you will definitely help our boarding scientists, research scholars, faculty members, and many more. Thank you, sir. Thank you once again.
Thank you
Professor Joseph,
also
I
for
just want to
the
say a
opportunity.
few things. Ki, I think this Artocarpus odoratissimos blanco, that one belongs to Professor left. Yes, Professor, have you left?
Come again,
was this?
sir.
Left, no?
No, sir.
No, sir, he is there, sir.
He's there. Okay.
Come again.
So I think that one belongs to the family of jackfruit.
Yes, the family Moraceae. Okay.
Moraceae. That jackfruit also comes under that one. I hope so.
Yes, both the jackfruit and the Artocarpus odoratissimus, which we locally call it as marang, they belong as well to the same genera, the genera of Artocarpus.
And the seeds of jackfruits also having that kind of activity, that is anti-diabetic activity. Is it there? I think whatever I can recall now, because in India, particularly in Orissa, from the land from where I am belongs, nowadays food processings are going on from the floor of that seed. Government is taking initiative so that it will work better for that anti-diabetic activity as it can know me, whatever information I have collected. And you also doing the work on that another species, that one is Artocarpus odoratissimus. A novel work.
Thank you for
sir.
that very informative premise, sir. Thank you so much.
Thank you.
Thank you, sir.
Yes, in the next session, we are going to meet Dr. Ajay Semalti, sir. Khushbu ma'am, please do the needful.
Yes.
Formalities, whatever with us.
Now, this brings us to the last plenary session of today's conference. I would like to invite our next speaker, Dr. Ajay Semalti. Good afternoon, sir. He is a course coordinator of India's most popular Swayam MOOC Academy writing, and he is working as a faculty member in the Department of Pharmaceutical Science, HNB, Garhwal University, Srinagar, Uttarakhand. Sir has 17 years of total academic research experience and more than 18 years of industrial experience. Sir has been included in the world's top 2% scientists of pharmacy and pharmacology by Stanford University Research 2020. Sir has 81 papers, 11 books, and 3 patents in his credits, and he is highly cited researchers with high index 26. We welcome you, sir, on the second day of International E-Conference. Thank you for coming today. Now may I request you to kindly lighten the session with your words.
Thank you. Thanks a lot, Khushbu. First of all, I would like to thank Professor Sahu for providing me the opportunity to interact with you people. And it is my second chance to interact with the Arkajan family. It's indeed a pleasure to have the discussion and with my fellow professionals, because in the journey of the online courses or development, I have been going multidisciplinary, interdisciplinary to almost to the every nook and corner of India and many countries. So thanks a lot, Arkajan family, for this wonderful conference, international conference, and the family and the counterpart, your counterpart from San Pedro College Pharmacy Department, Philippines. I also congratulate you people for coming together in this kind of collaboration. Without going for any further delay, I would like to come directly to my point, that what is the role of biopharmaceutical properties, what we can do with the biopharmaceutical properties of synthetic and herbal drugs in the pharmaceutical research. In the journey of research, in the academic and research, this is the kind of research which can be done in almost any kind of scenarios, any kind of resources. So I have been exposed to the some of the state-of-art facilities in the research and academia in some places of or the moment of my life of academic. I cannot compare the environment of labs in Japan or any other country or any other institute with yours and my college. My focus will be to trigger the students and the research scholar and the young faculty members to do the best out of the scarce resources. And that is the utmost need. With the availability of high-end instruments and high-end facilities, everyone can do the research. There's no wonder. But this is the hurricane task, that can you do the research in the minimum available resources or the scarce resources? Because in any institute, maybe you can't have each and every instrument and facilities available under one roof. Let me discuss today's topic, and I have already shared in chat box a link of further links. A document link is there. I keep on updating, so you just keep it with you. This is a link, tiny.cc/links by Simulti. So I'm going to, for today's lecture, friends, welcome from HNB Gadwal University. And I'm just going to full screen it now for you. Hold on. This is the Central University located in the lap of Himalayas near the bank of River Alaknanda. When you move from Rishikesh to Badrinath, it's just a midway around. It is a small town. With the blessings of Lord Badri Vishal, Baba Kedar and Maga, I welcome you all in this discussion in the International E-Conference on Natural and Novel Pharmaceutical Research. I'll be talking on the improving biopharmaceutical properties of synthetic and herbal drugs in pharmaceutical research. And my focus, as I already told you, it will be focused in triggering these future researchers rather than showing my endeavors. This is my this was going to be my plan of action. Okay. So NEP and research, biopharmaceutical properties, improvement, characterization support from various institutions, and research work with synthetic and herbal drugs. This is going to be the plan of action, National Education Policy 2020. It focuses on various topics, and what I want to focus, that no hard separations between the boundaries of the subjects. There are two things, multidisciplinary and interdisciplinary. We must focus that we can't make cocktail of each and everything every time if we are just talking about the multidisciplinary, interdisciplinary. The relevant, the rest with full rational logic, then collaborate with the holistic approach That multidisciplinary kind of research is the need of the hour, and NEP 2020, National Education Policy 2020, has made it the foundation. And doing the outstanding research is expected. However, I'm vocal about this thing, that outstanding research can't be done without the outstanding support from the employer, from the institute, from the nation. No silos. The most important thing is the breaking the barriers, overcoming those barriers of communication and collaborations, in which the departments are not communicating with each other, schools are not communicating with each other, institutes are not communicating with each other, collaborating with each other. That thing must be overcome, and this is the focal point of NEP 2020. As we focus about the pharmaceutical research, theoretical, computational, experimental, or multidisciplinary. In a gross, if I classify it, there may be many more things. Maybe you can add on the things like translation research, clinical research, so on. But I'm talking about the academic research, which are feasible in the, our academic environment. Okay. Now, what are the challenges? Scarce resources, silos, less interdepartmental, institutional, or academic industry linkages, lack of funds, support, willingness to do the hardcore research. These are some of the challenges. I'm not going to the detail of these things, otherwise this will, open up a new discussion. But I want to show the employers, the institutes heads this data, the research and innovation funds. The spending on research and innovation percent of GDP is just 0.7% in India. Researchers per lakh population is just 15, or just only 15 researchers. And total patent application, if you go, 45,057. It is the survey of 2017-18 done by the economics. It is the Economic Survey of India. While the China invest 2.1%. That's why they are having the 111 researchers per lakh population. And they are able to find this much 13, this much number of patents, multifolds. USA 2.8%, Israel 4.3%, small country. Researchers per lakh 825, and patent applications 6,419. Means that the total investment in research and innovation has declined from 0.84% of GDP in 2008 to 0.69% in 2014. And less than 0.5% of Indian students pursue PhD or equivalent level of education. So this is my message for the persons, if administrators are sitting over here, and if my message can be conveyed through the deans, through the shareholders sitting over here. Until you give the funds, research can't be done. The outstanding research can't be done, simply. So support the researchers first. This is my very first message if you want to promote the research. Recently, the 50,000 crore budget in NEP 2023 has been planned, and the draft has been finalized in July 2021 to boost the research and innovation. The National Research Foundation is being established. Hope the things will change. Now coming to our core topic. As you know, the biopharmaceutical properties play a very vital role in improving or modulating the drug delivery. We know the oral injection is the most commonly employed route of drug delivery. Poor solubility and low permeability of drugs leads to low oral viability. We know the if we are having the poor water solubility, the high doses may be required, chances of ADR may be there, safety issues, the solubility and permeability put together, their interplay is very crucial in both in the case of synthetic as well as in the herbal drugs. Various techniques are adopted. We know, we study in the text: solid dispersion, solid deposition, micronization, use of surfactant, use of salt form, complexation, etc. I have been associated, or I have been doing the work on the complexation technique specifically for last 2 decades of my academic career, and focus my efforts to improve the solubility, permeability, and sometimes modulating it, not only just improving, sometimes decreasing the solubility, or the improving the overall biopharmaceutical properties, solubility, permeability, dissolution of poorly water soluble drugs. And we can do the complexion with lot many methods, lot many we can base material we can use, but I have focused my efforts, research work on the phospholipid complexion and cyclodextrin complexation. Now, as we know, the phytophospholipid complex, however, the phytosomes are the trademark of Indena. Unlike liposomes, they are bound together, which with good stability and good bioavailability. They provide the good bioavailability of drugs which are loaded in the form of lipid complexes. Rather than the vesicle form, we are forming the complexes there. Simple things. Okay. Silymarin, silibin, silidinin, and so many things. Even some components in the terms of lipid complexes are there in the European market available. The Indian have been doing a lot of work, and many other companies are following the suit. Especially the flavones, xanthones, terpenes, anthraquinones. They can be converted into lipid complexes for improving the overall drug delivery or the bioavailability. You know, when I talk about herbal drugs, Ayurveda, our ancient or alternative traditional medicines, we need to work on those things and standardize those drugs, formulation, delivery system in such a way that they can be validated in terms of national and international standards. Then only, and it is the need of the era, to take the Ayurveda, to take the components, quote unquote. It's not just about that you can take the extract and do the things only. No. That's why we have been lagging behind in the international herbal drug research scenario, though we have a huge wealth of knowledge on that. But that translation is needed. Various kind of characterizations are needed, specifically the solid state characterization. Some of the things are available in your institute, but many things like DSC may not be there. Thermogravity, like TGA is there, microcalorimetry, IR, Raman, solid state NMR, Mosbaer, neutron scattering, so many things. We can take the help of these characterization support. As I told you that this is my message, that how we can do the research in minimum possible resources. Yes, we can improve the solubility. Okay, we can check the solubility. We can check the melting point, change in the melting point. When there is a complex, there will always be a change in phase transition. So we can detect it simply, maybe little bit information may be drawn just by determining the melting point. You can do it in your lab. Even the UG student can do. We can see the change in solubility. This thing we can do. Solubility can be determined. At least UV is there in every institute. But if we talk about the, sometimes in the herbal component, we need the HPLC. If you go for the, many students are having FTIR. Even in my institute, I'm not having the FTIR. So I have to run for the evaluations just for doing the FTIR. Now, if you want to see the changes in the X-ray diffraction, other things, you need the support from, get the support from Indian Institute of Technology, IIT lab, ISC lab, CDRI, SAF, sophisticated analytical instrument facilities available there, which is on the minimal charges. It is available. Instrumentation centers of various universities provide it. Just explore. Don't surrender that, yes, we can't do, we are not having anything. We are not having anything. Explore for the international collaborations. You are lucky enough that you have already got the collaboration, at least you have started in terms of that thing. And the research, you can take this collaboration to the research level in the future time. UGC DAE CSR, UGC Department of Atomic Energy and Consortium for Scientific Research Centers are available there in the Indore, Calcutta, Mumbai, and Kalpakkam. These are the major centers. Another center, the accelerated facilities available there in the New Delhi. So this is the platform. I'm coming that about the research. I just want to help you out that where to get the help, where to get the support. There is a website, csr.res.in. Just go there. You will get, you may get the user form there, or nowadays it is the online format is available there. Online proforma is available there. You just fill it out, and whatever kind of facility you want to avail in a particular center, you can ask for the help. You will have to give the details about your samples. Rather, the students are given the TA and the hostel facility to live there. You can't send the samples there in this particular center. However, many IIT labs and all that, many centers give the facility for sending the sample by post. So Indore, in Indore Center, including RRCAT, Raja Ramanna Center for Advanced Technology, Indus Synchrotron Beamline like MOB, HRXRD, LTHMF, low temperature, high magnetic field, okay? DSC, ESC, SCM, TM, many facilities are available. Just go there in the website, you will get the thing. Mumbai, inside the Bhabha Atomic Research Center campus, neutron refraction, neutron scattering, DLS. In Calcutta, HRXRD, LTHF, radiation chemistry, gamma radiation facilities available there. DLS is available there. In Kalpakkam, again, the HRX, XPS, NMR. What I mean to say, don't limit to the melting point and solubility. Do further characterization, contact the centers, go talk with the scientists, talk with the collaborators there. Take the help of your mentors and explore them. Then you will be able to conclude something concrete out of your work. Okay. Now we have recently done a workshop on the CSR awareness workshop. It is with the help of CSR conducted it rather, and we hosted that workshop in our institute. You will get all the details. Just note it down, swayam@hnbgu.in. You will get a navigation bar there, CSR awareness workshop. We have given all the lectures, videos there, available links available there. You can get the things. Because when we want to see the changes in the material, actually we are entering into the domain of material physics. We will have to learn the proper characterization. It is not like that preparing a cyclotron complex is some kind of outdated topic. Yes, it is outdated if you are just claiming that solubility increased by this cyclotron complex. It is but the demand of the topic is you go deep inside the topic, explore the molecular interaction, what is happening there. Don't leave the topic unexplored. We are doing this silly mistake just by giving the excuse that we are not having the facilities. The government of India is giving you the facilities for free. No fees charged there. We got first research support from UGC DAE CSR Indore in 2003-04 and got a good publication, though it was on the case of some simple SCM figures that took free of cost to go. Now, lipid complexes. We prepared for some synthetic drugs like diclofenac sodium, diclofenac potassium, then let's say the aclofenac and aspirin. So we prepared this. We started that we wanted to work on the herbal drugs, but we started the prototype or pilot study with some synthetic drugs, which we know already and which we have worked on various things, simplest kind of molecules, just to set up the pilot study. Now, these, when complex with the lipids, their properties are changed. Now, why solubility is getting changed? Why melting point is getting changed? You have to prove it correlated with various parameters like DSC, XRD, SCF, but not go for anything which is not required. Maybe I remember that I have a freeze dryer or lyophilizer in my lab, in my project lab. A researcher came to me, sir, I want to get the lyophilize my product. I thought, so why do you need it? Just I need it, it would be better. If you can't maintain this lyophilize product in that condition, it will obviously get some more moisture from the environment and will be damp again. Justification is very important. Discuss with your mentors. So what you are seeing in this here, then DSC thermogram, if you can, because when we want to go for phospholipid complexation of a particular compound, you have to create some connectivity. So that's why we acidified the diclofenac first. From the salt form, we convert into acid form so that it can form the complexes with the lipids. Dipalmitide, phosphatidylcholine, different kind of lipids can be taken. We use the 80% phosphatidylcholine because if you use the 40% grade, it will be sticky kind of thing. If you use the 90% or above, it will be very hygroscopic one. So we used 80%. It was safer to add up. We found that, as you can see here, it is a published study in the, I think it is in Acta Pharma. In 2009, we got it published. In 80% PC, if you say this is the drug, this is a lipid, and in the complex, the endothermic piece is clearly shifted. It's indicating that, yes, what is going on inside the complex. And same, the change in trypsin peak was found with the APC. It is again a published paper. With the aspirin we tried. Now we compared this scenario with the HRXRD. The diclofenac, they think, especially you can see that a drug is having this sharp endothermic peaks. These peaks you will see there. Sorry, crystalline peaks, not when we're talking about the X-ray. Basically, X-ray diffraction is providing the crystallinity, information about the crystallinity. So diffuse peak you can see here in the case of complexes in the diclofenac, as in the similar in the aspirin, similar in the aclofenac. We have done each and every thing is published there, and then the different ratios we can try. We can see the changes in the surface morphology when we are changing the drug to CD ratios or kind of lipid or lipid percentage. We can see the changes and correlate them with their profile with the solubility. As you can see with the diclofenac, we found a diclofenac PC complex around double solubility increase. Aspirin, you can see this change to 8.45 to 15.98. Aclofenac, again, dissolution study. So simply, dissolution, every pharmacy institute is having the dissolution apparatus. Just compare these things with the dissolution profile for these drugs. So when we got these hint from these synthetic drugs, we took the herbal component. Mark my words that we used the component directly, the pure one. We brought it some from Sigma, from other companies like TCI, Tokyo Chemicals. Gallic acid, naringenin, quercetin, catechin, these are the flavones, major flavones. Gallic acid, we worked upon that. Again, we found the similar change in crystallinity, which led to the improvement in solubility. Although the gallic acid has already got the solubility, but because it has got the simpler structure, we tried that. We tried it because we could understand what is going on there in the at the molecular level, or you could at the what kind of interaction is happening there. Naringin, again a flavone is there. Unlike the phospholipid complexation, cyclodextrin complexation is a little bit different. I'm taking care of my time, don't worry. Just in 45 minutes, as started at 2 to 2:45, it will be over. With 10 minutes remaining, I will give you the discussion time. When we go for the cyclodextrin complexation, it's a geometrically governed complexation, basically, because it's basket kind of thing, which has got the different specific dimensions. Cyclodextrins, which are cyclic torus shaped oligosaccharides with 6, 7, 8 alpha, beta, gamma CD cyclodextrins. These are parent cyclodextrins, and some are analogs of these cyclodextrins. The cavity, how the molecule is entering into that cavity is to be understood. That conformation and numbering, or I can say, if you go for the conformation of the cyclodextrin, this is a smaller opening and wider opening there. And it gets wider from alpha, beta to gamma. Gamma has got the most wider opening. We can add some component to improve its complexation. Hydrophilic excipient, it may be like nicotinamide, PPPK30 we can use. We have also published in I think letters in drug design discovery, Bentham Science, acyclovir, we studied on that. And so we again tried those things with the FTIRs, again the DSCs, DSC curves, X-ray diffraction for naringenins. Almost each and every, I think nothing is uncovered, nothing is unpublished. Every work, every figure is published here in the reputed journals. So dissolution study, we can compare the dissolution, you can do the comparison, but remember that, can you prepare a calibration curve through the UV spectrophotometer to select the component or the drug wisely so that you can analyze it UV spectrophotometry if you are having only the UV spectrophotometry. If some component can't be evaluated just by UV spectrophotometry and it is there is a need of HPLC, then you need the HPLC either in-house or you can go for exploring. But exploring HPLC for analysis is quite typical because it requires time. Phospholipid, nemosalide, we also tried the similar kind of comparison with the nemosalide. We tried for improving its, how notorious nemosalide is regarding the water solubility after the amphotericin B. So we tried the same, compared the lipid complexation as well as the in the that is CD complexation, compared their profiles in each and every aspect with respect to dissolution, solubility, and compared these properties to draw the conclusion that what is the dominating factor and what are the things which can affect the complexation. What are the parameters? Now, again, I'm emphasizing, this is not my aim to show my research. That's why I'm focusing on each and every graph. This is the way you can do it very simply. You can go it with that. It looks heavier, but it is not. All the things you have, you can get it done. You go with your samples to the institutes, get it done, but sit with the scientists, with the collaborators who have got the knowledge of interpreting these things. And obviously, you must have a very good literature survey. With nimesulide, we got very good release when we go for the cyclodextrin complexes. And we saw that what is the impact of complexation on different class of biopharmaceutical class of drugs, class 1, class 2, class 3, class 4. I'm not going to the details. You can also check it out that can you take this cyclodextrin complex to the nanoparticle level? I'm not going into the detail of what does the nanoparticle mean, but what are the characterization parameters you go with for the nanoparticles? If you are starting a study, see that can you land at a place where you can draw some conclusion. With the help of SDS, we tried cyclodextrin complexes. And this is a figure from a paper in which we hypothesize that how the amphotericin molecule should enter into it's a molecular docking study or molecular modeling study. It's a published paper of a scientist. We tried that how it is entering into the cavity. Can it make the difference into its property? Yes, it makes a difference. So we planned a project in which we tried to quantify the interaction between drug molecule and cyclodextrin and location and configuration of drug within the cyclodextrin with the help of small angle neutron scattering. That too, do you know, this is the pride of India. These are the two nuclear reactors of government of India, our nation. The one you can see here is the Cyrus, the round one, globe one. It is decommissioned now. And the second is the Dhruva, which is working as a research reactor. So I was lucky enough to have, I'm lucky enough that I got the opportunity to work into this, into the lab inside the research reactor in the beam line of that neutron beam. So neutron scattering we study. This is the neutron beam line study where we study the neutron scattering. We just learned something. I'm not saying that because it is a core physics. When you go multidisciplinary, you will have to learn the other aspects of other subjects, other topics also. Then only you can conclude something. DLS, phase solubility study. It's very simple. You can perform the phase solubility study in your simple laboratories. And then you can correlate this data with the particle size, zeta potential, with dynamic light scattering. Though DLS is also very expensive equipment, I think DLS equipment is not less than 50 lakhs. So we have the facilities there. Then we correlated these things with our permeability. Because acyclovir has got a permeability issue. So we studied because cyclodextrin also increases the permeability. The profile of scattering, neutron scattering, which we got, interpreted that. Then we found or correlated that yes, it has formed some spherical host-gas interaction house with a certain radius, which we identified there. This is the kind of thing which is required. You can't end up the research just increase the solubility. This much time, this much time. No. You have to quantify the things. And this is the work which the samples which were analyzed in the PSI, Switzerland lab, what the drug and amphotericin one. I'm not disclosing the thing because we still have to publish the results. But as you say, the orientation of the curve, that neutron scattering, gives the hint about how the interaction is happening and what is the you can say what is the shape of that micelle or molecule. Especially it is very useful in the nanoparticle research, but a very few get the chance to get these analytical facilities available because they are the mega facilities. But you know, there is huge scope on material physics, but we have to work on need to know basis only. So we compared it with our acyclovir nanoparticle permeability. Again, we got a very good paper, and recently, I think day before yesterday, we got a very wonderful paper in the Journal of Inclusion Phenomena and Macrocyclic Chemistry, in which we studied the photostability effect of cyclodextrin, which we conducted with the doxycycline. It is recently published. You can see in the Inclusion Phenomena and Macrocyclic Chemistry. Yes, some drug is having good solubility, good permeability, but it's having some photostability. So, you know, the CD complex, the cyclodextrin complex, can also prevent the photodegradation. It can improve the photostability of the component. Sometimes what is happening that we are not having even the compound to work on. So just make a bank of the drug, and with a good justification and the rationale, start working. What is my key message for researcher? That do some preliminary work. You must have a good rationale of the study, problem definition, pilot discussion with scientists, rational planning, experiment, samples, user form you must fill. You go there, get it done. After doing the preliminary study, work schedule, progress, sample planning, and discuss the experimental results. This is utmost important. And this is our work which we could do with these facilities and available out of career, and 84 papers, maybe, yeah, around 11 books, 3 patents, out of which one patent is granted and 2 are filed. E-content development, and I'm lucky enough to get this thing that yes, we have got this course industrial pharmacy, which is very useful for our students. So these are some of the list of publications out of UGC CSR facilities. You can get it from any profile there. So we have got our work done with some Swayamprabha modules also. So I acknowledge UGC DE Consortium for Scientific Research, Bhabha Atomic Research Center, Mumbai, PSI Switzerland, Panacea Biotech, Baddi, and the entire team. And I would like, just before ending my presentation, I hereby present to you the Pioneer MOOC in Pharmacy by us, by my team, for your student, teachers, and researchers and industry personnel. It is the MOOC on industrial pharmacy by my team, which is ready for on the SWAYAM platform, which is going to be started again on the January session. And this with industrial pharmacy first, strictly on the basis of PCI syllabi. And we are lucky enough that we are the first one. We have created the character journey. 11,500 learners across 100 countries we are having. Do join us. It is available on the swayam.gov.in. Okay, with good hands-on videos, laboratory environment, lab setup live interactions, unit operations directly, or you can say eye-catching engagements. So I welcome you all, and this is our recent endeavors out of the course that we have come up with the 3 books. So you are most welcome, and the links are available. Please go ahead there. And another MOOC, for which I'm having more than 46,000 learners across 150 countries. Academic writing, especially for PG students, researchers, and faculty members to learn how to write the papers. Tiny.cc / AW 2022. The link is given there. Just join us with a promise from my side. We promise to trigger an excellent academic writer. Okay, so please do join us. It's by free to enroll. Enjoy learning with us, and we have got the national and international coverage there. 6 courses were selected from the SWAYAM, and we were there in the top 30 online course in 2019. Again in 2020, again in 2021, we were in the best online courses of the world. So join us. It is also available for online. The links regarding some aspects of the academic writing, open education resources, SWAYAM, Arpit modules, and a podcast available there. Let's just explore it. 4 courses for the students are available in the form of Swayamprabha modules on novel drug systems, pharmaceutics 1, cosmetics, and pharmaceutical production technologies. The links are already given there. Tiny.cc/links by 70. Okay. And in some private edtech platform, some free courses are available there. Tiny courses, bigger courses are available, some free, some paid. Just enjoy it and enrich your knowledge. The people who are crazy enough to think they can change the world are the ones who do. If you want to achieve something, you have to run for realizing that the benefit out of that research, and don't forget the societal contribution. I thank my mentors, UGC, CAC, MRC, and Ministry of Education, Government of India, to join Industry Pharmacy MOOC. And I think I would have been able to inculcate something so that the research endeavors can be triggered by the students. Thank you.
Thank you, sir. Thank you, sir, for this wonderful session. Anyone have any question? Please ask.
How, sir, you people made that industrial pharmacy, that new book is opened, no? So, and you are also giving the that MOOC presentations for the e-learning kind. So whatever the machineries we are going to taught for the students, how those things you bring?
We have given the links there.
Do you get my point, sir?
Yeah. Actually, we cannot mimic the entire course in the form of book. Yeah, we cannot mimic. For enjoying a course, you have to enroll in the course. They have got the different segment. The book cannot replace the importance of the course, and course cannot replace the importance of the book. So both have got the independent features. So I think that's why we have given that. If some people are having, because after doing the course is closed for them, though you need not to go to again and again. Yes. So what we have given, at least you will have a further reading links and important links which we will be giving there with the books and all that. And in the form of book, you will get it a ready reckoner with you people, with the students, and we have written the book keeping in view the students' mindset, that how easy we can make it to reproduce from the learning point of view or from the representing point of view for the exam point of view. Thank you. Otherwise, there is no scarcity of the books. You can have n number of books.
Yes,
but
yes.
What is easy to understand, that is important to bring.
Yes.
Thank you.
Like it, sir. Yes, Khushbu ma'am.
Yes, thank you,
As it
sir.
is the time of lunch.
Now I would request to our Dean Sir, Jyotirmay Sahu, to kindly felicitate Ajay sir with token of appreciation for his contribution in today's training session.
Thank you, Ajay sir. And also I would like to thank Mona ma'am. Last day I was a bit engaged with otherwise, so I cannot remain present at the last moment of the that e-conference of yesterday only. And for that, I am sorry. And please accept the
Thank you.
memento.
I humbly accept, and thank you for your cooperation, for giving me the opportunity to shift my schedule. It was inevitable for me. So that's why I requested you. Thank you for considering my request. Namaskar.
And the memento will be at your home with post.
Thank you.
Thank
we
you,
will
sir.
meet you
Thank
soon.
you very much.
Namaskar. Thank you, dear participants. Feel free to connect at any time for any kind of academic help. Thank you. Bye-bye.
Thank you, sir. Now, I would like to request Dr. Bhavita Sarangi, Associate Professor, School of Pharmacy, Arkajan University, to give vote of thanks. Over to you, ma'am.
Yes. Now this is the time for vote of thanks. It is such an honor for me to get the opportunity to thank our distinguished speakers, Ranjit Bharaskar sir, CEO QBD International QBD CGMP Consulting United Nations Advisor Geneva, Dr. Nagaraj Rao, Managing Director, Rane Rao Reshumya Laboratories, Navi Mumbai, Dr. Ajay Samalti, Professor, Department of Pharmaceutical Sciences, HNB, Professor Joseph, Faculty Pharmacy Department, San Pedro College, Philippines, Dr. Ajay Kumar Thakur, School of Pharmaceutical Sciences, Delhi, Pharmaceutical Sciences and Research, University, New Delhi, India. Thank you so much, sir. Thank you for inspiring and encouraging us with your kind words. I would like to thank our Dean, Dr. Jyotirmoy Sahu, Akka Jain University, Jharkhand, India, and Professor Fatima, Dean, Pharmacy Department, San Pedro College, Philippines, for their outstanding contribution in organizing this 3 days e-conference. My special thanks to the coordinators, organizing committee members, faculty members, and all the technical assistants for their cooperation and support towards making this session successful. Last but not the least, I would like to thank who are involved directly or indirectly for making this event meaningful and successful. Thank you all. Thank you.
Thank you, ma'am. Now I request to everyone to please stand up for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you everyone for your presence. Now we will
can you request all the participants whoever here to unmute their video to give a screenshot?
Yes, sir. Please, it is a request to all the participants to switch on your camera.
Please, all of you, unhide yourself so that we can take a screenshot. Please help us to do this. So whoever is there, please do the needful. Okay, अभिषेक अभी आप ले सकते हैं स्क्रीनशॉट, जितने लोग हैं उनको ले लीजिए.
Thank you, everyone.
Okay.
Thank you everyone for your presence in today's plenary session. Now we will meet after a short lunch break at 3:20 p.m. for our scientific session. Thank you. Thank you, sir.